Yes, I got it. And large portions of its nervous system can be imaged. I must confess that presentation by Zoom, I'm not my cup of tea. So they are really well suited to relating neural activity to behavior. And more specifically, what our colleagues in the neuroengineering lab do is you have this brain, and there's something interesting, which is called the ventral nerve cord. It's kind of, roughly the equivalent of a spine. It's where you have these descending neurons, and then you don't have too many of them, 700, that go from the brain itself to the part of the fly body that control behaviors. And they are believed, they don't control pose directly. What they control is behavior. And then there's more machinery further down to actually control 3D pose. So when the fly behaves in a particular way, you should see some of these 700 neurons light up. And because there's not so many of them, it becomes manageable. So here's what it looks like, where you have the eye, which is stuck to a plate, and it's on a kind of a treadmill. The treadmill is a ball, and it's acting on the treadmill. It can move. And on the left, you see your video data, and on the right, you have what you get with so calcium imaging data. You use 2-photon microscopy and a lot of magic that I do not fully understand to generate these images of the activity of these neurons. And here's another example of the same thing. So what our colleagues have done is they have these long sequences in which they have annotated behaviors. So you see 8 different behaviors. So walking backwards, grooming, which is this, and other behaviors that the fly can do. And if you categorize them, here's what you see. A histogram of the different behaviors and when the fly does them. Okay, so far so good. The difficulty starts here. These are 4 different flies doing the same behavior, and the images of the neural activity is very different. So if you do standard supervised training on this, it doesn't work too well. It doesn't generalize well from one individual to another. And this is really the problem we've tried to tackle in this work, is can we come up with a representation that will generalize across individuals? So that's the challenge. And so, of course, if that being the case, you need a lot of annotation, which is no good. And an additional problem, which we don't typically have in normal vision, is this calcium imaging has a decay. What you see in one image is a weighted sum of what's happening now and what happened in the past. So you are going to have to handle that. And our proposed solution is we've gotten fairly good at estimating 3D pose from video data. That works more or less. So can we use that to help supervise and come up with better representations? And the benefit is what we aim for is close the domain gap between individuals, I should have said instead of images, and handle this problem of calcium decay. Okay, so as I said, we've become reasonably good. In fact, it's funny because it goes back to the very beginning of the computer vision field. This is Newbridge, 1878. So in that, I follow my thesis supervisor. The oldest reference I can stick in the presentation, the happier I am. So Newbridge did this in 1878. That was the very beginning. And of course, things have evolved since then. And I think Hedvig this afternoon will tell us what's happened 150 years later. But, well, horses are fun. They're big. And flies are kind of smaller. So the techniques we're going to use are not going to be that different, but they have to be adapted. So this we did. So in the lab at CPFL, what we have is a setup with 7 cameras in which you see the fly behaving from different angles. And from this, you can find interest points, you can compute the 3D pose, and you can get the 3D pose information. So here's what the setup actually looks like. So it's pretty involved, but it works. And you have this poor fly that's stuck to a plate on its circular treadmill that's in the middle of all this. And for those of you who are computer science PhD students, I think you should thank your lucky stars. You realize that your colleagues in neuroscience spend their times breeding these flies, cleaning the mess. And in fact, my colleague, the one I collaborate with, when a new PhD student shows up, gives them their very own tube full of flies that they have to deal with. I don't do any of that. My students don't do any of that. We just deal with the images. It's actually a lot easier. So here's a setup, the schematic of what it looks like. You have the 7 cameras, the fly in the middle. And what we did then is in fact relatively standard, which is we train an algorithm to find the joints, to find the key points. That's the middle box here. And once we've done that and it's robust enough, it's kind of an active learning kind of thing where we run it, we make mistakes, we correct, and we do it again and again until it's reliable. And once we've done that, we just do stereo. And when you do stereo with 7 views, it works. So it works, but of course it's cumbersome. I mean, this is not a setup that anyone could have. And which is why we also work on single view 3D capture. And to do this, it's essentially the same idea, except we have a setup where we have 1 cameras, but prism, so that you get what's called a ventral view and a side view of the fly. So we get 2 views at training time, which you can again do stereo, get the 3D, and train the network to lift 2D joint detection to 3D. So we use the ventral and side views for training purposes, but at inference time we use a single view. And in fact, it's a little less accurate, but it's comparable and it could be used too. So if you feel like experimenting with fruit flies, you can. So now for the heart of what we're going to do, given that we have these synchronized, and that's important, we have the video data, and at the same time, we have the calcium data. They are not at the same frame rate, but they are synchronized. And use this to learn representations that are useful and that we can then use to classify behaviors. So what we are going to do, so we have this behavioral data, which is the 3D pose data, the neural data, which is the calcium imaging. They are synchronized, and we are going to use contrastive learning to actually learn to send that to a latent space of lower dimension. So which is what's called Z on this slide. And this is again relatively standard, but the key to actually making it work in this case is to do the right kind of augmentation. Because we want something that's going to ignore the identity of the fly, because we wanted to generalize. So we are going to find a way to make it ignore the identity of the fly. But we are also going to need to make it ignore these artifacts due to the delay in the calcium imaging. So we will encode and predict it to latent vectors, and in standard contrastive learning fashion, we will try to maximize similarity when the behaviors are the same and maximize dissimilarity when they are different. Okay, so we'll here same thing. We have a loss function that we, which is the 3D pose. We have responding calcium imaging, it's called positive. We have a negative, which is a different behavior. And we are going to write a loss function based on normalized cross entropy. That is so that when you go from the images to the Z to the latent vectors, when the behaviors are say they are as similar as possible, and when the behavior is different, they are as distant as possible. And if you just do that, what you're going to do is bridge the gap among modalities, so between video, calcium imaging, but you will not bridge the gap between animals because it will use the identity of the animals in the Z vector. The trick here to make it be identity agnostic is to do what I'm calling here a swapping implementation and training time. So suppose you have the those data for 3 different forms. And what you can do, so it's essentially a trajectory over time. And what you can do is in pose space, you can look for animals that have a similar pose. And you replace it, so which is you leave it for a second. So you can see that this blue dot here has become green. This guy instead of this guy. To build mixed trajectories with different flies, so different identities, and that's what you're going to feed to your contrastive learning algorithm. So that will take care of identity. Other thing we have to take care of is this calcium decay. And you can see here on this thing, these are recordings of the raw data. You have the actual behavior as labeled as fairly sharp transitions. And then you have what you see in the neural decay, which is, it corresponds, but it's smooth and a bit shifted in time. And that's because of this lag. So you have to make your deep net ignore that. And the way we do this is we add to our neural data a signal for an additional neural data where we explicitly add decay so that it learns that these things that decay ought to be ignored. And we also blend different neural activity images because, in fact, when I say a pixel lights up, well, it can light up because 2 different neurons fire at the same time, which is not that good. So we also have to teach it to ignore that. So as often in machine learning, well, the technique is standard, really. The key is to give it the right data so that it learns the right thing. Or it learns at least what you want it to learn. And it actually works. So, for example, this is the representation of, on the left, the raw neural data. So the colors correspond to different flies doing the same behavior. And of course, we are not the first ones to do this sort of things, but the other people were not using the pose data. And when you represent the latent encoding, you kind of see it's still kind of clustered by identity. Whereas our result on the far right is not clustered, it's really well spread. And to a large extent, identity has really been removed from these Z vectors we compute. So that's what our goal. We've closed the gap in between individuals in the learned representation. Yes.
Do you so this is with a set of individuals.
Yes.
Have you evaluated then this on a new individual that wasn't seen during
Yes,
that?
yes.
So it does it still end up kind of in the same?
Yeah, I think actually this on this specific slide, I don't remember if that was on the testing set, but yes, we of course we have a testing set of individuals that were not used to learn. Actually, that's what we did here. So another way to test this is to use this representation for performing some downstream task, which is action recognition. So that definitely is done on non-used individuals during training. And something that's interesting, so we have the usual table. And something that's interesting is, as I said, the supervised learning techniques, which are in yellow, are terrible on this. They really failed badly. Then the other people who, there's really a need for specific algorithms to do this. And incidentally, this is not limited to flies. I mean, we took flies because my colleague that's what he does. But this works also for humans. So, for example, we've tested this approach on a different kind of data. And it's available on the web. You put electrodes on a skull, a human skull, and I think in this case you're dealing with epilepsy patients. So you put these electrodes, you record them, and at the same time, you take images. In this case, it's 2D, not 3D, but it's 2D, and you record their motions. And in this case, there are two kinds of behavior, either at rest, if they have epilepsy at rest, may not be static, but they are not actively doing something, and actually moving. And so that's one data set. And we've also played with some favorite data sets where, in this case, this is not multimodal because it's all video, but we pretended it was multimodal by saying each of the different cameras is a different domain. And so the goal here is to make it ignore perspective effects. And again, if we try this and compare to what you get with other formal representations, we get better results. So in conclusion, this is promising. It shows that 3D poses extracted from video can be used for unsupervised learning of neural representations. So that's good. As I said, it's promising, but it's not good enough yet. And okay, so if you look carefully at the numbers, our numbers are better, but they're not. You know, they are not out of this world. I'm aware of it. And why? So that's the question. And I think our current hypothesis is that what we are doing is too coarse. It's not a problem of the machine learning per se. It's a problem is right now we are looking at a single slice of that fly. So what we need to do is to model not a single slice, but a volume to get really the extent of the neurons and to get a better resolution. And that's essentially this. And maybe next year I'll have something to say about it. And of course, where do we want to go with this? Well, it's almost as scary as Neuralink. I'm referring to this as the Frankenfly project. The idea is if we understand it well enough, we should eventually be able to stick an electrode at the right place and make it do something. So that will not be next year, but we've been fine. But that's where we and that's it. Thank you very much.
Yeah. That was confident, inspiring talk. First of all, I wanted to say I hope we all appreciate how easy it is to work with standard data sets and files. So we'll be taking questions from this room. Please, come here. We'll also be taking questions from Zoom. So if you're over there, feel free to post them on chat. We'll be reading them.
Great talk. Question about, so when you were showing that it was hard to classify across flies, the different action categories. Okay. So across flies, it was hard to classify actions, right? Because of this, you know, and perhaps some of the individual differences are at that level.
I don't think we could with this. So the question is, have we looked at classifying poses? And the answer is no. I don't think it can be done with this particular data because these neurons, I mean, when I'm doing this is really the equivalent for the fly. And these neurons do not control pose. Essentially, you send a signal, and then there is very clever machinery below that decompose that signal into individual poses. So we would have to record much more than what we have.
Yeah, I think I understand that point, but what if the action categories are too coarse? The action categories are too coarse, and maybe across flies.
So the action category is mostly walking forward, walking backwards, what they call group flies, this kind of motion. And a few more are the most prevalent ones when I've seen.
Ask one quick question. So you used the images for the second part, right? For the process learning, you were vetting the images themselves, the raw images. You didn't segment out the neurons and get traces. For the second part of the talk, you were using raw images, right? Did you consider
raw video and calcium images? So it's two photo images.
Do you think there's individual differences in level of morphology that you could just segmented them out and just dealt with the traces?
Part of the plan.
Okay.
And that's what I said is if you just do it in 2D, you're not feeding enough information to the thing. So really we have what's next.
Thank you.
Anthony Newts from Kitware. You have these two modalities, like calcium and video. Seems like the calcium is very, very messy. So it's very individuated responses to neurons from different individuals. So I imagine you did experiment. I'm not sure if it was in your table, but did you look at what level of recognition you get just from the calcium and just from the video, and how well the fusion of the two helps the two?
Essentially, the numbers I have in the table, the other just calcium.
Nice.
So that would be what you can do. So we gain something like 5% with N% will be from better calcium imagery, 3D calcium imagery.
So do you think from the neuroscience perspective, is the issue and noisiness of the calcium an artifact of the imaging, or is there really just that the different neural patterns for the same action varies this much based on individual?
Messy. I mean, actually, the flies aren't individuals, so we have to deal with. Which is actually, I mean, now I'm going entirely or out of my sphere of expertise, but I have been told that and when you look at its strategies, they're different. So they really are individual. So this is inherent to the beasts. And of course, the resolution is not what we hope for, but that's essentially that's the best that can be done today.
Also have a question on Zoom. That's a question from Sophia, and she's asking, for action recognition, what kind of architecture did you use when learning the features for the contrastive learning?
Essentially, standard convolutional layers followed by projection layers, and nothing. Details are in the paper, but I mean, nothing that you couldn't. We didn't use transformers. Guilty.
You said for a single view, which view did you use? Like I guess the side view, like the bottom view. I'm kind of curious.
The bottom view.
Oh, the bottom view. I see.
Yeah, because we want to see
I see.
the legs. Really, when I say behavior, it's really it's really the legs and a little bit the antennas, but you see all of this in the bottom view.
I see. Can you say again about the activities again? What kind of activities they were? Like how many kinds of activities?
So this poor fly, okay, you have to imagine this poor fly, its back is stuck to a metal plate. So one fly. And so it's on a rolling ball, so it's like a treadmill. And so it can go forward, it can go backward. I've often seen this like a bit like a cat. Grooming, it's called grooming for a reason. It's like a cat. And there are a few more that I forget.
And then they're labeled by hand, I guess.
Yeah, we can go back to the slide if you want. So because they're here. So here as the okay, so what they are called. So resting is one, of course, it's not doing much. We have walking, which I've mentioned. Antennal grooming, which is here. Abdominal grooming, where okay, so I must confess, I'm a computer scientist. So to me, that I never looked in too much details at what they really do. And of course, one of the dreams of my neuroscience colleague discover behavior, because all these behaviors that have been classified by people. It would be super cool to discover a behavior that we've never seen before, that the thing would come up, oh, this is a new behavior, but haven't.
Questions in the room, we can still take them. We still have a couple of minutes. In the meantime, I was wondering, what's your kind of roadmap from here? Like, do we need to study flies for some years to come, or do we need to consider other animals? Like, what degree of generalization would you need, and how would you expect to scale this setup?
I think okay. So let's take the question the other way. One of the favorite animal of people who study neuroscientists is a worm. It's C. elegans, 137 neurons. And we know all the neurons. We still don't understand how it works. As far as I know, we know the wiring diagram, we know everything, and we still really don't have a clue as to how it works. I mean, if somebody, again, I'm not an expert at this, so somebody is welcome to contradicting me, but so C. elegans, 137 neurons, we don't have a clue. Has 100,000. I think it's going to keep us busy a few more years. So of course we are doing mice and monkeys and people, but before we really understand, it's going to be a long time. And I mean, I'd like to share actually, it's a sad story, but it's worth sharing. One of my other colleagues, his wife was suffering from, he's a neuroscientist, and his wife was suffering from clinical depression. I mean, serious stuff. And then when he said that, talked to the doctors and they never understood what she had. And this guy is a top class top flight neuroscientist. And the gap between what a top flight neuroscientist can do and understanding why his wife had the disease she had, the gap is huge, immense. And I'm not sure it will be bridged in our lifetimes, but I've been wrong before. Thank you very much.
Thank you very much, Pascal, once again. So our next talk is going to be remote. I don't know, Meredith, if you're here, please give us a sign. We're a little bit early, so we will need to see if Meredith has joined.
Joining from Mozambique.
Yeah, Meredith could not be here today because she's in her research camp in Mozambique doing field experiments. So that should be really fun. But first, I don't know if she's here, and also there can be some connection troubles. So in the worst case, we'll have to take a break if she's not able to join. But fingers crossed.
Are a little bit early.
Oh, yeah, 10 minutes only.
We are a little bit 10 minutes early. So.
We can take more questions for Pascal. I'm sure this room is excited to know more, so don't be shy.
Can I ask a question? How did you start with fly people? That doesn't really go, I guess.
Start working with fly people. I think well, serendipity is the school offered, our university offered some funds for interdisciplinary collaborations. And there was this neuroengineering lab who needed some expertise in machine learning. And that's how it happens. And actually, that's one of the reasons we are very, very fortunate, because I don't only do flies, I also do planes and a few other things, because in the end, the tools are the same, but they are applicable to all these different fields. So.
Are your students eager to work on this type of topics, or, how do you manage like the science and like the vision aspect of this thing?
Well, it's fun because I can expound on topics I know really very little about, but because I talk to these people, I learn. So that's a very, very welcome change from, trying to do 2% better on this old boring benchmark. Yeah, I'm getting a little tired of it. So that's a way to keep things fresh, because there's new knowledge, new expertise, new stuff. And so that's one part. And on the other hand, it's actually not that difficult, because as you've seen, for me, all of this is an image. An image is an image and an image. I mean, if it's a fly or a plane, in the end, it's going to the same transformer. So with, of course, specific tricks. Something that makes it interesting is, okay, I'm not a theoretical machine learning person. I'm not really able to contribute that much to the theory of machine learning, but what I can contribute is domain expertise on how these, what we want to do given the domain knowledge. And it's important, maybe PhD students, is that even in the age of machine learning, domain knowledge is good. And what you already know, there's really no need to learn it. Just find a way to structure your deep net so that it takes advantage of that. And that's essentially our chief scientific activity. And that applies to anything I'm talking about.
I have more questions actually. Oh, there you go.
Yeah, so great talk. I just wondered, you mentioned about the 3D representation, generalizable, and I understand, but activity recognition and action is just still cognitive, so like in two-dimensional representation, video sequence, and so on. So, for that case, like yeah, I understand that we some certain viewpoints. So get to that kind of viewpoint. So and also in the animal domain, there are a lot of people who are using house. They still use like 2D representation. So what do you think about? So can you comment on this having the more information from the 3D representation? How that's more useful for the 2D representation?
These later presentations a lot is to impose priors. That's the way, I mean, and a lot of what we do, for example, when we do 3D shape modeling, priors on what we want to model. So, I mean, there are many other users to be sure, but that's the one, that's the key one I'm aware. Does that answer the question?
Right. So I just wonder, like, what? So what if instead of using 3D representations, what if you just use like 2D representation? I mean,
What kind of representation?
like 2D key points from the sequence of the 2D key points. It's your representation instead of 3D.
Seems to help. The representation helps.
Yeah.
If we fed, that's why I mentioned this strictly supervised method that failed. So my impression, it's not a proof, but my impression is it would not be as powerful. In some sense, this distillation process really finds some, if you do it right, finds what's essential about it, what's invariant, and that buys you something.
Actually, that was related to my question to ask somebody who doesn't. And it's nice that you're using these methods to lift like 2D key points to 3D. Yeah. And that like you use these 2D key points, 3D key points that it's predicted from 2D. But if it's from the bottom up view, I'm kind of questioning how much the predicted 3D actually helps.
There's a lifting network. So we've trained, either we get 3D direct from the multiple view, okay, no problem, or we look from the bottom, we see the edges of the fly are below. So from the bottom view, we see them relatively well. They are the occasional, but the lifting, that's why you train the lifting network. It learns how to ignore these occlusions and gives you decent results. Has to do, I mean, that's part of the answer to the question is they are strong priors is, I mean, the motion of all these limbs is not independent. I mean, all the degrees of freedom are connected to each other. So if you see a part of it, it's enough to guess the rest in general.
Yeah, like actually how to train these things.
The scale of the fly?
Yeah, how many, or I mean, like how many images? How many images? The images number.
2 millimeters.
How about number of images to train this lifting network?
These long video sequences with hours of stuff. Which actually is the point of having this if somebody had to annotate everything, forget it. Radically with 3D pose estimation is important.
How is that? That's so great.
Maybe that's not very relevant question, but I noticed that you mentioned 100 neurons on the brain to control high-level activity of the fruit fly. So I'm just wondering that does that mean that fruit flies don't have the ability to like just like human to like control the motion, like pose very precisely or whatever. Control it.
So to but what my colleague has explained to me is that these neurons give the command walk forward, and then there is machinery below that decomposes this command into the exact poses that need to be more neurons. I mean, more neurons further down. Like we have our spine is part of our nervous system. And if I go back, oh, I don't have the slide anymore, but if you look again at my slides, I have the brain, the ventral cord, and then something below whose name I forget. That's where the machinery is. Additional neurons that will essentially decompose that into the individual poses.
Research since they are not so like
So I'm not sure, to be honest, I never asked a fly, but I don't know that a fly can say, I'm going to move my little finger by 2 degrees just because you asked me to. But again, I don't know. So we would have to ask a real biologist what they can and cannot do.
yeah, definitely.
Thank you. So let's see once again if Meredith has joined. Meredith, if you're there, please say something. Sound. Are we getting sound through the laptop then? Yeah, but I mean for her, the sound for remote talks would be from the laptop, and then we just move it. Yeah, that's a good point. Okay, I don't think we have any indications that she's here, so. Okay, organizers, what do we decide? Should we take a break then?
Yes, we're gonna take a 40-minute coffee break.
Yeah, so we will regroup at 11 in this room for Sarah's talk, and we'll see if we can suggest Meredith and somebody else. Thank you.
Also, let's thank Pascal.
Oh, yes, the one that.
Yeah, it's like I had to give a talk, and like the Wi-Fi was like and it actually ended up working out. It was like I didn't even want to be, and I had to turn the camera on.
So I want to ask, we should, I'm in the second class. Should I?
Your poster on itself is red. We don't really need to swap the whole poster. We can move on to the poster. Yeah.
Me first.
But we are sorry, so we, yes. Yes.
Yeah, okay, thank you very much.
Can we go to the slide? Is this Hyunso's? Yeah, CV4Animals. Here we go. Because I want to put the schedule up. Um,
I see.
I want to share this screen with the people so
Yeah.
that when people come in, they know that it's
Yeah. Okay.
coffee break.
We have monitor.
Oh, by the way, we're planning a dinner actually. Shohei and Natalia. Tonight.
Tonight?
Yeah, if you could join. I don't really know the details, but we have a joint dinner with which both of us are supposed to be. Here, let me mute this actually. Here, let me just share this. Share. Actually, hi. Sylvia, actually, I want to check for later. If Sylvia can hear me, because it's like, if, oh, hi, Sylvia. What are we gonna do when the speakers are remote, like Hedwig, Fig later, and like their voice, like,
I think that if we start the talk, so the speaker is this speaker is loud enough, I think.
So first of all, maybe you can stop recording because
yeah,
then
yeah,
you have
okay. I'm gonna
minutes
stop
of-
recording for that.
Yeah.
Yeah, that is I think those are yeah maybe.
How do I stop recording? Here we go. Pause.
Oh,
It's better.
Okay.
I don't know why mine is echoing like that.
Can you speak?
Hello? 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 Can you hear that on Zoom?
Yes. So his mic is not working, right? Working. Okay.
yeah. Okay.
Hey, everyone on Zoom, can you hear this?
Yes. Yeah. Good. Thank you.
Okay, it's time for us to resume the session. So please, everybody take your seats. Our next speaker is going to be Sarah Bewie. Sarah recently finished her PhD at Caltech, where she worked with Pietro Peronna, developing methods to improve global scale biodiversity understanding, including work on wildlife monitoring from camera traps, community science data, satellite data, and sonar. Her work is highly interdisciplinary, and she works directly with stakeholders in the field to deploy her tools and ensure they are both usable and useful. She seeks to break down knowledge barriers between fields. She founded a successful BI for conservation Slack community with other 900 members. And she's the founding director of the Caltech Summer School on Computer Vision Methods for Ecology. And today's talk is going to be about participatory human BI elephant population monitoring. Sarah, over to you.
Thank you. Yeah, so I thought it would be fun to talk a little bit about, to this audience in particular, that mostly focuses on specifically the computer vision models themselves, about everything that goes around a computer vision model when you're actually deploying it in the field, and the ways that we can actually work directly with stakeholders to make sure that the models we train and the papers we publish kind of translate to impact and are actually directly usable. So I'm going to be specifically talking about one case study, which is a system that we've built that actually leverages human intelligence to help improve robustness of individual identification of elephants in the greater Mara ecosystem, alongside the Mara Elephant Project. And this is work that definitely would not be possible without an amazing team of collaborators. Peter Kulitz is a student of mine from Caltech who's actually been living in Kenya for the last 4 months, working on expanding this system. Arjun Subramanian has been working with me on social network analysis. Jake Wall is the director of research and conservation at the Mara Elephant Project, and Wilson is the leader of our long-term monitoring team, here down on the left, and they're all amazing biologists from Kenya who have been directly working with the elephants and with the data collection processes, and then Chuck Stewart from RPI and Shane Lubold from UW. And so just as like a, I'm sure some of you know where the Mara is, but in case you don't, it's this transnational region between southern Kenya and northern Tanzania that encompasses the Mara Triangle, Mara North Conservancy, Lemek, a bunch of different local conservancies on the Kenyan side, as well as Serengeti National Park on the Tanzanian side. And it's very famous for being sort of the location of what's termed the Great Migration. So you can see here, you know, hundreds of thousands of wildebeest actually migrate from Kenya to Tanzania and back every year. But they're not the only things that migrate, and elephants migrate too. So the elephant population is a transnational one, which means if you want to work with this elephant population, you actually have to cross national borders, which becomes interestingly and politically complicated when you're thinking about wildlife conservation. And there is a pretty large elephant population there. It's at risk, but it's relatively stable, around 2,500 individual elephants. And the Mara Elephant Project, who are our collaborators in the area, they've been in operation since 2011. It's a pretty small scale organization, but they're really massively focused on conservation technology, incorporating really data-centric and data-driven and technology-driven approaches to try to mitigate elephant population loss. And it's around 75 people that all live, I don't know if you can see it here, but this is the conservation area. And, well, it's not fenced, so there are elephants walking around, mostly breaking water pipes. I actually just got back from Kenya like a week ago, and there was because of COVID restrictions, I had to sleep in a tent, which I actually love, and I could hear and smell the elephants walking past my tent, which I was like, don't step on me. So the good news is, since the Mara Elephant Project has been operating in this area, poaching threats have been going down. You know, in 2011, there was a significant amount of poaching happening with this elephant population, but through education and mitigation efforts by them and many other organizations in the area, we are seeing a decrease in poaching, which is really good. Though unfortunately with COVID and with some of the complications around, you know, ecotourism no longer being super financially viable, there was actually an uptick. It's not in this plot, but 2021, we saw some growth in poaching, unfortunately. But the thing is, just because poaching is going down does not mean that that the animals themselves are not at risk. And so I'm gonna go through a few of the different ways, in very complex ways, that that elephant populations and wild animal populations are having to handle changes in climate and in human encroachment on habitats. So the first big thing is that we're seeing massive deforestation in the Mara. So here you can see in red, that's all of the forests that have disappeared since, like basically was lost between the years of 2000 and 2018. Most of that loss is due to clear cutting for agriculture or illegal charcoal burning. And down here, in the lower plot, you're seeing actually the amount, the percentage of forest loss that we've seen just in those last 15 years. And a lot of that, there's also a huge amount of habitat loss, not just in the forest, where savanna has been moving towards agriculture. So what you can see, all of the yellow is now agriculture. You can see it's really surrounding and starting to encroach into these traditional conservation areas. There is a lot of overgrazing. So depending on the conservation area, they do or do not allow livestock grazing within the area. And so what you're seeing here is This is what's called albedo anomaly, but it's essentially the sense, if you take a mean in 2010, and then you look at the difference for these different conservancies in terms of the amount of green versus brown grass, where the brown is where it's been overgrazed and then so essentially dies. You see big spikes, and it's actually, you can see that the two conservancies, the Mara Reserve and like the sort of large scale conservancies where, to give some context, the Masai Mara, the tribe, the Maasai, owns most of the land, and a lot of these conservancies, the tribe actually still owns the land, but they get paid rent by conservation areas to protect the ecosystems. But as part of that agreement, they are allowed to graze their livestock on their own land, which makes sense. But this is where you run into the fact that humans and wildlife are often in direct conflict for resources, water and grazing and food. And so the conservation areas where there is grazing allowed, you can see there's been massive decreases. Yeah.
A very naive question. Elephants live in forests.
Yeah, and the savanna, both. And actually, so there's savanna elephants, and there's also forest elephants in Africa. And savanna elephants are slightly larger. We're talking mostly about savanna elephants, and I'll be talking about savanna elephants. But the forest elephants, they're smaller. They're a little sneakier. They're very quiet. They're difficult to detect, and there's not a lot that's known about them. But the forest elephant populations are in much more rapid decline across Africa than the savanna elephants are. Yeah.
What is illegal coal burning?
Oh, so people cut down trees and then build these coal kilns and burn them to make coal, which is then used for fuel. And it's illegal in many areas, but yeah, difficult to catch. Yeah. Oh, yeah. So I forgot, I didn't mention the desert elephants, but there are another species, another type of elephant in Africa as well. Thank you. And so then as you start to see this loss in food coming from overgrazing, from loss of habitat, what we end up with is a lot of human-wildlife conflict around crop raiding. Elephants are pretty smart. People's agriculture is like delicious and lots of good nutrition. And so the elephants will actually learn to work together to break fences, to make sort of concentrated attacks into people's farms and eat their crops. And what you see, especially because most of these farms are small scale agriculture, really small holdings, a group of elephants can come in and completely devastate someone's livelihood very, very quickly. So this is active conflict. And what this means is that people will actually then kill elephants, not to poach them, but to try to protect their livelihood. And it, unfortunately, elephants will learn to do this and then teach other elephants how to do it. So you'll have kind of these specific bad actors that are repeated crop raiders, where they're actively teaching other elephants and increasing the amount of elephant population that is raiding crops. What you can see here are tracks where they've categorized normal behavior, and then crop raiding is all of the red, where you can see that the elephants are doing these raids into agricultural land and then coming back into the conservation area. And as a result, because crop raiding is an issue, we're starting to see more and more fencing across the Mara. And these fences that are being built in more and more robust ways. And what they do is they limit corridors. They make connectivity between the actual protected areas more and more difficult. So this increase in fencing affects not only the elephants, but also many other species of wildlife. And so the more fences that are built, the less connectivity there is. And then that ends up being really restrictive in terms of elephant migrations, access to food, but also genetic diversity. And so this is actually, again, looking at the total percentage of fenced area and where that's changed from 2011 down to 2020. Yeah.
Are fencing to protect people or keep the animals inside?
It's fencing to keep the wild animals out.
I see.
Yeah. And a lot of it is electrified. Yeah, so that's a lot of different things that are threatening wild animals. And I'm mostly talking about elephants, but also there's an incredibly diverse ecosystem of large mammals that still exist in Africa, and all of them are being affected by a lot of this. And I don't think that we can really make the call that what we should do is we should just only keep this land for the animals. I mean, there are people that have like ancestrally, this has been their home, indigenous groups for hundreds and thousands of years. So it's not the case necessarily that we can say, no, okay, everyone just needs to leave. We're going to just keep protected area for only animals. I think we need to start thinking about how we can build systems that enable wildlife and humans to coexist functionally. So there are different ways that people try to collect data to understand how the population is changing, to try to detect rapid change for the negative or the positive. One of the most common and most traditional methods are these aerial censuses. So they fly low-flying aircraft, and you can see these like long sort of grid-based searches, and they're actually counting just human. They have one human flying and one human in the back, looking out the window and counting how many elephants they see. This gives you raw population counts, but to be able to fly these surveys is very expensive and honestly dangerous. These small planes go down a lot. One of the largest causes of death of wildlife biologists is like aerial censuses, which is pretty sad. There is a huge push towards automating these things. So moving towards, drone-based censuses. And I think that's really exciting. But it's still really cost prohibitive. So highly labor intensive and expensive. And so usually that means they're only doing them every 5 to 10 years. And it's also only giving you counts. It's not giving you any more nuanced information about the demographics and definitely not about the individuals. So the next thing that people use to monitor are collars, GPS collars they put on wild animals, elephants included. And this gives real-time movement information, which is an incredibly nuanced source of information. And a lot of people work with Earth Ranger, which is a tool that is hosted by the Allen AI Institute. And essentially what Earth Ranger is doing is it's allowing conservation areas to keep track of and manage all of this different real-time movement information. So, Mara Elephant Project, their Earth Ranger, they track all of their ranger vehicles. They track their long-term monitoring team. They have a conservation helicopter that they use to actually push elephants away from agricultural land. It's like an interesting mitigation strategy. They also, when I was there, we went up to find an injured elephant and report back to the vet to see basically what was injured about the elephant. So it had an arrow in its back leg, and we were able to tell the vet where the elephant was, and then they could go trank the elephant and give it veterinary care. So they track all of their vehicles, but then they also track the 25 elephants that they have with collars. And they're often collaring big tuskers. So these are elephants that have very long tusks that are at high poaching threat. Now, this is amazing. You get this really nuanced, almost real-time information about animal behavior, where they're moving over time, but for 25 individuals. So they're very invasive. They cost 25 grand per collar. And the collars die. They need to be recollared. It's really difficult to actually scale up an individual monitoring using this type of information. So one of the things that people do to try to monitor at the individual level is visual re-identification. And this is where all of us come in. This is a computer vision problem. You're looking at images of animals and you're trying to identify in a very fine-grained way, not just what species are they, but what individual animal are you looking at? And there's been amazing work that's shown this to be highly effective for patterned species. Essentially, you're looking at a fingerprint. And so with things like the zebra, Tanya Berger-Wolf, who's here in the audience, has done amazing work on with a nonprofit called WildMe, where they actually are able to identify individuals of how many different species, Tanya?
64 and counting.
64 and counting in different species, where they can use their patterns to actually robustly identify individuals. And this is great because this means that you can do what's called visual mark recapture. You can use traditional ecological modeling techniques to understand statistically the global population by looking at resighting rates of individuals just using images. Very non-invasive. You don't have to tranquilize the animal. You don't have to fly your helicopter with a tranq gun out the side and put people at risk. But, it's easier for animals that are easy to identify. So elephant re-identification is possible with a lot of expertise. This is a very old paper from Elephants Alive. where they're showing these manually drawn ear contours that were being used to identify individual elephants in Kruger National Park. So they had binders full of these, where they were explicitly drawing the contour of the ear and recognizing individuals based on that contour structure. And this is interesting because these things are stable-ish. Ear contours can change, right? But they can change only in certain directions. So in this case, you can see from 2007 to 2013, mostly the contour of that individual was the same, but they gained a new hole in the ear. And this individual on this side, they gained a new sort of big notch in their ear. And so what they're doing is humans are learning with expertise and mostly relying on these ear contours to be able to identify individual elephants. There has been some work looking at the wrinkles in the face, the vein structure in the ear. There's other characteristics that are actually also quite identifiable. If an elephant is missing a tusk or one is significantly broken, there's one elephant that its tail got eaten by a lion in our population, and he's called tailless, and he's really easy to find. So then what this has slowly turned into is then, trying to capture not just those ear contours and not just looking at these drawings and trying to match them to a binder. People have slowly looked at different ways to try to build coding systems for individual elephants. This is all coming from the pachydermy literature. And so Joyce Poole originally had this very nuanced system that was looking at these different ways that you could delineate different types of ear contours. And then recently, Michelle Henley developed what's hopefully like a little bit more novice-friendly system for coding. And so what they're doing is they're looking at they turn an ear into almost like a clock. So you have these different sections of the ear, and then you're marking the most notable features in each section. And then they also have presence of, missing tusks, any other like significant features. And these coding systems, mostly previously managed in Excel spreadsheets, were still enabling humans to identify many more elephants than they could just trying to keep it all in their brain or in binders. And so what they've turned that into is this thing called SEEK, which it's termed the System of Elephant Ear Pattern Knowledge. And they have a data set of this from Kruger National Park in South Africa going back to 1996. And essentially it's tracking gender, age, tusk presence, positions of primary and secondary ear holes and tears, and any extreme special features. And it's a very simple vectorized system. You just get basically one code and then you're looking at similarity. And what we found is that the experts there they're aware of the ways these things can change. And so they have sort of in Excel, like they'll put in, question marks for unknown features, and they'll start putting in question marks in different places to see if they get better matches into their system. And this is where we came in, and we were like, okay, I think we can do better. And so there had been some previous work, also mostly with a student of Chuck Stewart's, looking at trying to take a contour-based matching system that was originally built for recognizing individual whales based on their fluke contours and adapting that to elephant ears, which are much more flexible and tend to have much more complex backgrounds. It's not like a fluke against the ocean. And they were able to do pretty well with something they call curve rank, which is basically a wavelet-based system. So they first extract the contours, and then they take different representations of that curvature, different sort of smaller and larger segments of it to try to build some scale and variance into it. They build a representation of the contours, and then they're doing matching based on that. Unfortunately, it really struggles with angles, occlusions, and blur. So as the image quality gets worse, these systems become less and less reliable. And also, unfortunately, the Mara Elephant Project and the Mara population in general had no data. We had data from South Africa, from a completely different population, but there was no way for us to successfully use that system on that database of individuals for the Mara population. So what we wanted to design was a participatory approach. We wanted to build something where there was a system where our collaborators could immediately start to monitor their population. before the computer vision is good enough, and slowly start to build up more and more robust computer vision systems and start trusting computer vision more and more as it's proven and vetted with strong evaluation. And so what we did is we started to really heavily rely initially on this seek-based coding system, on a data collection procedure and a system that enabled people to, even non-experts, identify individuals through this code. But in the process, collect the computer vision annotations that we need to be able to build up our computer vision training data over time. We wanted to make sure that our system relied heavily on human verification, because this is a high-risk endeavor. If we start misidentifying individual elephants, we'll start getting massively incorrect assumptions about the size of the population. These are numbers that, you know, the Kenyan Wildlife Service is depending on to make policy decisions about conservation. So human verification is important. Think about it like healthcare. You also, you know, we need human verification for healthcare decisions as well. We wanted to slowly reduce human effort over time. So the initial amount of human effort was high, you know, coding in these manual attributes, building these seek codes for every individual that's seen. We wanted to slowly reduce reliance on that. And then we really wanted to work directly with scientists in the field to deploy these systems and learn from the deployment. You know, not just build up a benchmark data set and get results and walk away, but actually really vet our systems in the real world. And, you know, we wanted to also build on their existing monitoring systems, their existing workflows, and their existing data management. So what we did is we worked with that Earth Ranger dashboard, where they were already collecting group sightings of elephants. And we built sort of an API to API communication so that the information could be ingested from Earth Ranger to our system, which we called Elephant Book, and then ingested back into Earth Ranger, so that you can actually now in Earth Ranger track individuals based on collars, but also based on visual re-identification. And then the way our dashboard works is essentially you first go out and collect data in the field. Currently, this is based on our biology partners going out and driving essentially routes, transects, where they identify any individual that's seen along that route, and these routes are repeated for, you know, statistical viability. But we're also now working to incorporate camera trap data from the WWF Biome Health Project in the area, and so investigating how much difference in image quality affects our ability for humans or our automated system to ingest individual IDs from images. And then, you know, they go in and they add a group sighting to Elephant Book. So that means you take in something from Earth Ranger and you say, okay, yep, it was this sighting reported by, you know, whatever. And then they upload photos for that sighting. So now these are individual photos. And the protocol we use is that they go out in the field, they see a group of elephants. They try to photograph one individual at a time. So they'll take multiple images of an individual, trying to capture each ear really clearly. And then between individuals, they take a picture of the sky. And so that way, you know, it's easier for them when they come back to make sure that the images of an individual are mapped together. So it's like a very, very simple system to try to keep data quality in control. And then what they do is they go and they box images of individuals across the group. So we have a system where now, if you have, you know, 4 pictures of that individual, you box the individual in each one. And this means now we have, you know, nice, clean, like this is individual A. Here they haven't actually matched into the database. We're just relying on the context of a short time window to say, these are all the same individual in the context of this group. Yeah.
The sky, can you explain why you do the sky? Is it for lighting or actually?
No. It's literally just a separation flag. It's just saying like, hey, you know, because if you've gone and you've seen 7 groups of 150 elephants at a time, yeah, you go back, you're like, oh, which ones are which? And in between groups, they take 3 pictures of the sky. You know, I mean, stuff like this works.
It's good actually for lighting estimation later.
Yeah, yeah, exactly. You know, I love it when things can be reused. And then what they do is for each individual, so then all of the sort of boxed examples get ported into a new window, and now this is termed an individual sighting, and then they manually code these seek attributes, and they only code what's visible in these different things. And then that's where computer vision comes in. So now we do right and left ear detection. We extract those contours from curve rank. And then we also have been experimenting with some more of these, deep learning approaches, these metric learning based. And it kind of has to be metric learning because this is not a static population. I think that one of the most important points about monitoring a population of individuals is that it's always changing. You're always having new individuals be born. And we still are working our way up to the entire population. We've covered around 1,000 individuals now, but there's another 1,500 that we're still going to see. So we're looking at incorporating and ensembling in some of these other deep learning based approaches. We've been seeing some pretty good results from something called pose invariant embedding that was originally a facial re-ID system. And then there's this verification step. So we show them the most likely matches and they human verify based on matching into representative images in our database, which individuals been seen. So from the computer vision side, what we found is that you can't trust the computer vision alone. It's not nearly good enough yet for something this difficult, but it can make human experts more efficient. What we can see is that you start getting better and better top one accuracies. So, top 1, top 5, like the actual individual that you're looking at is somewhere within the top matches you see. And the closer it is to the top, the faster it is for a human to verify that match. And the interesting thing is that the seek-based coding alone gets you pretty close, but then actually incorporating computer vision does better than just seek coding or just computer vision. So these ensembles of like human intelligence and vision get us closer to something that's really usable. And we have been seeing these results slowly improve over time. And I think there's actually something quite interesting about the fact that I don't have the results in this slide because it's still like actively being explored. But when we're incorporating some of these metric learning approaches, we find that they do significantly better on cases where these traditional things like these contour-based, wavelet-based matching, which feels like old school computer vision, but honestly for an open set with very, very few examples per individual, they're still pretty robust. The really interesting thing is that they do tend to really learn different things. And this is something we're really trying to understand is like, what is it about deep learning versus these statistical approaches that's fundamentally different? What are they focusing on that's different? And how do we build more and more complementary systems by bringing in all of these different types of feature extraction? And then one of the things we've been looking at recently, which is really fun, because it means I'm having to massively expand my expertise and spending a lot of time looking at graph theory and network science, is we're trying to figure out if we can replicate human experts, which is usually a good thing to try to do. So we found when we were talking to human experts that they were really heavily reliant over time in learning social information about elephants. So what they would do is they would see a group of elephants, and then maybe they'd see that same group of elephants later. And there would be one elephant like Flopsy Lopsy, who has very floppy ears that they really easily identified. And then they'd be like, oh, well, Flopsy Lopsy always hangs out with Bob, so that's almost certainly Bob. And then they would check and they'd like, yeah, it's Bob. So the social context was being really heavily relied on. And this is something our systems did not have. So what we started trying to do is now that we have, we deployed this originally January 2021. So we have a year and a half of social network information. Now what we're trying to do is construct a social network from these historical resightings and then figure out if we can leverage that network to contextualize individual re-ID in sightings of groups of elephants, and figure out if we can combine IDs of known elephants in the group with co-sighting probabilities from our network to enhance that re-identification accuracy for unknown elephants. So yeah, again, it's similar to the process ecologists use in the field. And this fun graph is like over time, the way the social network of elephants sort of grew as we collected more and more sightings. And, this is a great idea, but there are actually a lot of challenges. Part of it is that if you look at the number of sightings per individual, you can see that the vast majority of individuals in our database have only been seen once. Now, once you see an individual one time in a group, we think that already should be informative in terms of this social network. But if you've never seen an individual before, It's not going to do anything for you. And you know, there's a few individuals. Fred is a big tusker who's been seen 17 times. He has amazing tusks. They almost like touch the ground. And he just is a really, I guess, a very social dude who likes to hang out where the long-term monitoring team goes. But so there's all of these other ways that the network that we learn just based on our co-sighting matrix, which is just like elephants that have been seen in physical proximity to each other by our team, the way that that network is unstable. So one, elephants hide, particularly depending on certain times of the year. What you'll see is a group of elephants near the edge of a forest, and you can't actually see into the forest to see which, so you're only sighting partial groups. We also have issues because elephant social networks are not simple, they're complicated. They're four-tiered hierarchical systems, according to, you know, elephant social network biologists. And so depending on availability of resources like food and water, they'll either glom into these huge megaherds that are controlled by a single matriarch, which is often the grandmother or the great-grandmother of everyone in the herd, or as resources diminish, they start splitting apart and they end up in their family units is like the smallest unit, which is just a mother and her calves. And those calves, you say calves, but they tend to hang out with their mom until they're 12. So subadults also. So you'll have elephants that look quite large, but they're actually still very young. And so then you have transient bulls. So the female social networks are stable, the males are not. There's inconsistent data collection patterns depending on different users. They might have different policies around this. And there's new individuals all the time. And one of the things is that the co-sightings themselves lack granularity. And so what we've been doing is we've been building this social graph, trying to incorporate spatial and temporal context. So each node in the graph actually has metadata around the space and time that it was collected. And we try to incorporate metadata using like IESEX, using these expert mediated rules. And so there's a few methods we've tried here. We first tried to do strict family assignment and then use like an ILP-defined optimization system to try to maximize over that. What we found was a disaster. There was way too much sparsity. And you know, we really didn't have nearly enough information to build these strict, this is a family group and this is who belongs to it. So then we started looking at greedy assignment based on visual matching scores. So essentially very, very simple. You just say, who's the easiest to identify individual in the group we've seen? Okay, now based on that, you want to now combine for the visual scores, the likelihood that each other individual in the group is an individual plus some weighted score based on the past social sightings. And then you just take the maximum over that and you iterate through. This actually worked a bit better, but there were significant issues if you made an incorrect prediction the first time, right? So then we looked at, a simple version of that, like beam search version, where you just say, okay, well, let's take the top 5 and then let's work down this tree and figure out what's possible. That actually worked a bit better, but the thing that we found worked best, which makes sense, was a more participatory approach, which matches the way our system actually works. So you give an expert the group of individuals, you give them the first one, and then you have them verify the match, and then you incorporate this social network to re-ID. What we found is that these approaches are improving over our baseline, these are micro-averaged over time. So basically it's like you take any point in time and you can only see the past sightings, and then you're looking at improving that social network score over time. The improvements are not as much as we'd hoped. And we really think that this is probably because of all the instability in our social networks. So we've actually now been looking at other social species that have more stable social networks to try to understand how these measures might be applied to say plain zebra that have very, very stable harems. And then, you know, just really quickly, like we built this system, what have they actually learned? So they've been able to see a significant growth in the individuals that they know about. They've been able to capture demographics of their population for the first time. They've significantly better understandings of where the elephants are distributed in their conservation area. They are starting to see multimodal monitoring. So these are sightings of an individual matched with the collar track of that individual. And for the first time, they've been able to start tracking conflict and injuries. So seeing how these injuries are changing over time, and where the locations of the most new injuries are happening. And so we're expanding the system into 3 new regions now: South Africa, Northern Kenya, and Tanzania. We're trying to incorporate multimodality. I mentioned the camera trap data. We're building more interactivity into the system. So we're trying to actually look at, instead of asking for all of this human expertise to code each individual over time, how do we build participatory approaches where the model queries the most useful information to reduce the uncertainty around individuals? And we're also looking at expanding into body condition and health monitoring. So how do you look at fine-grained 3D modeling of a single individual over time to understand how their health is changing? Cool. I went over time by a lot. So if anyone has questions, that's good. Otherwise, we should probably go to the oral.
Yeah, let's take one quick question.
Sorry. I looked down and was like, oh, crap.
Said that the aerial surveys were expensive, but these days with drone and automated tracking, is this still true?
Oh, yeah. Because drones are not cheap. And the ability to have drones that don't break, and people who can fix the drones, and people who legally fly the drones, it all actually ends up being very expensive. The hope is that these costs will slowly reduce over time, but as of right now, it's still incredibly expensive to fly drone surveys in Kenya.
Also not allowed in these cases.
Yeah, there's a bunch of restrictions around drones. Okay, well, yeah.
Just
Yeah.
quick one because we need to move on.
Yeah, I'll be around for the posted sessions. I'm super happy to answer questions then.
Okay, once again, thanks a lot for passing out. Now we will be moving to oral presentation, and the presenter is Matthew Dawkins. If you can start sharing your slides, that would be awesome.
You can just unplug my.
Okay, so the talk will be the title is just fish track technology and a sample data set for multi-object vector exploration. Matt, over to you. Let's see if we can hear you.
Can you hear me? Okay.
Thanks.
I'm sorry, can you hear me okay?
Yes, we can hear you.
Perfect. All right, so I'll try to keep this brief, but thanks for inviting me here today. My name is Matt Dawkins, and I'm a software engineer at Kitware. And I'm here to briefly discuss FishTrack, which is a new data set we've been putting together, which is a compilation of both imagery and annotations performed at a few other organizations, particularly NOAA Fisheries, the US, Ephemer in France, and the California Department of Wildlife. We have a couple end goals with FishTrack. The first is basically just multi-object tracking evaluation, where there's obviously a lot of challenges in this domain that I'll go through in a moment. The hope is that these techniques can improve population assessments of specific species using non-invasive camera surveys on a couple different platforms. Tracking in particular might provide some information that previous surveys don't have. There's other sampling metrics such as max N counts, which take the maximum number of target species occurring within some time interval that biologists use a fair amount. That value then gets extrapolated over multiple flex. There's lots of other things you could do with sampling schema for good tracking information available. It also lets you do additional measurement along these fish because you take multiple measurements along the track in terms of how large they are and then average those into a single discrete value. So that's its main goal. A secondary goal is comparing different detectors and classifiers, particularly those which might use motion information, which isn't required for all these data sets that we'll show in a second, but in a few cases, I suspect it will help a lot. Lastly, we want to train up new trackers to bootstrap annotations of new data sets. For example, biologists who are considering whether or not to use computer vision, who are sort of on the fence if they want to go down that route or not, or use other types of survey information. So a summary of the data set as it is now, we are looking to expand it over the next couple months, and additionally add in some new annotations from a few groups we're in contact with. But it's approaching about 1 million frames and boxes right now across a couple tens of thousands of tracks. We provide a pre-made trained test split just for consistent evaluation of tracking algorithms, which again is the main goal of the set. Has roughly 250 species of fish, but that said, not every single data set has labels at the subspecies level. So there's some generic fish or unknown fish labels in there as well. We're releasing an early beta tomorrow. Right now, about half the annotations are online. But then in a month, an updated release with a couple more tweaks and new sequences, and then finally about 2 months down the line, a final release, the challenge page. I'm going to start going through these slides fairly quickly just to get to the posters. There's a couple different collection platforms this data is coming from. CMAP is collecting a lot of data in the Gulf. It's a multicam system. You can see, the bars on the imagery, which divide fish in half sometimes. There's the mouse camera system, which is a drop cam. These are roughly stationary surveys, but you can see the imagery bobbing in the water, so they're not completely devoid of camera motion. Secondly, there's a lot of GoPro-based platforms. Some are baited, some are non-baited to attract fish. Again, the biologists use different sampling strata in each of these cases. I guess the two unique cases here are the California Department of Wildlife's camera, which is our only freshwater sequence right now, and the ephemeral trawling camera that you see right there. You can see the video playing. That's the only one with non-natural backgrounds right now, but we suspect that the same types of tracking techniques are applicable in this domain. So training jointly on them hopefully wouldn't hurt performance and would hopefully help performance. That's a secondary goal of this data set is determining when training together helps and when it hurts versus training on individual data sets. Imagine there's tons of challenges in this domain. One is just moving debris in the water, sediment. You get cases such as marine snow, other organisms sometimes. The other you could imagine is there's lots of schools of fish, especially when you have a baited system, they might all be attracted out at any given time. There's a huge juxtaposition in these data sets. Sometimes you have absolutely no fish for an hour. Sometimes you have, seems like all the fish in the sea coming to this camera at once. You can imagine there's lots of occlusion issues and some issues you don't get in traditional tracking problems. The other major problem is just a large degree of dynamic natural backgrounds. Sometimes the fish have very low contrast against the backgrounds, and really you can only make them out from a motion signature. Sometimes when they're over open water, you can see them a bit clearer. Lastly, when water condition breaks down, sometimes you just see fish disappearing into the background. They might not be present. And again, you might just see a subtle motion signature, which actually disappears and you can no longer see the fish. And then you see another blob emerge, which might be the same fish, might not. But also have a lot of very small moving targets that, again, you can only see sometimes with motion signatures when you zoom in very far into the imagery. Sometimes the biologists can actually determine their species. Sometimes they can't. They just have an unknown fish label. You're talking about this level of annotation. So most of these annotations were performed in a software toolkit called Viame that we've been working on, which has both web and desktop annotators, but we are again looking to include annotations performed in other toolkits now. Viame sort of has a couple of purposes. It lets biologists train up sort of models from scratch just by checking sequences that are fully annotated and training an ensemble of models like FastRCNN or depth-based detection models. Some of this is very general and can be applied to lots of different domains. Some of it very specific to this underwater domain where we might be doing some extra stereo measurement on top of tracks or image enhancement to improve the image quality, both during annotation and in processing algorithms. My last slide, I promise, but we trained a couple baselines on this approach. It's not actually on the full data set right now. It's on about half of it, but we have updated models that are in the works right now on the full data set. It's basically just a cascade FastRCNN detector with a separate appearance-based tracker on that tracking the intractable that fuses sort of kinematics and appearance into the same classifier that links individual detections. In terms of future work, one of the near-term things is adding polygons and head-tail points to these fish, both to support that measurement problem and improve the detection performance a little bit. We aim to do that with a combination of sort of automated and unautomated approaches with manual review. Lastly, we might add some additional ensembles from other work within NOAA, such as animals from the air, covering different pinnipeds on coastlines. This data set also primarily covers the tracking problem, but there's lots of data sets that just have individual frames of fish. So we might post a sort of a related fish ID tract on the same website. So if you have any questions, I encourage you to reach out to our poster. We'll be at both the in-person session after 12 and also the virtual poster session. Thank you.
Thanks a lot, Matt. It looks like a really fun data. So for questions, let's indeed respond them to posters. So this talk concludes our morning oral session. Now we're going to have 2 hours for the posters presentation, both in real world here on the first floor and online. Please try to have lunch at the same time. We're not going to have separate time for that, so you'll need to be efficient. And we'll get back here by 1:40 for the next round of talks. Thanks all.
Okay, maybe we can stop the recording.
Yes, let's do that.
Maybe it doesn't have to end now, but yeah, we can stop. Okay, so let's get started. We'll start the second session, the afternoon session, and our first speaker is Helvig Kjellström. Am I pronouncing correctly? Yeah. So I'm very honored to introduce Helvig. Helvig is a professor in the Department of Robotics, Perception and Learning in KTH Royal Institute of Technology, Sweden. And she's also principal AI scientist at Silo AI and affiliated researcher in the Max Planck Institute in Intelligence System in Germany. And her research focused on understanding human and animal behaviors. And Helvig has received a series of prizes for her research, including 2010 Poindring Prize for the fundamental contribution in computer vision. So with that, Helvig, please take away.
Thank you for this nice introduction. Happy to be here. So today I would like to talk about our research on recognizing expressions of pain in horses from video. So it's really important for the welfare of animals. that we can detect when they experience discomfort and address that as soon as possible so that they this discomfort is taken away. And so I will explain why we need to do automated recognition for this. So in Sweden, as probably in other countries, one of the most common causes for culling horses or killing them basically are orthopedic disorders in problems with joints, which are not lethal if you could find them earlier. So if we could detect signs of pain earlier, we would have much better chances of treating these horses instead of taking their lives. And even veterinarians are bad at this. It's really, really difficult, even for a trained professional, to see when a horse is in pain. And I mean, as in other areas, computers are generally better at systematically detecting weak patterns in noisy signals. And it's even more difficult because horses are prey animals, so they maintain a stiff upper lip, you may say. So it's basically beneficial for them to not show that any weaknesses. So it's really good. If you want to see their true expressions, you should observe them when they are alone. Yeah, so in order to achieve methods for automatic analysis of horse behavior, we have gathered an interdisciplinary team consisting of veterinarians at the Swedish University of Agricultural Sciences, and also we collaborate with Silvia Suffi, who is an expert in really detailed 3D tracking of animal pose and shape. And we have 2 PhD students working on these projects, Sofia Bromé and Si Li. And our collaboration also includes other groups. Yes. So an important way of finding out about injury and illness is to see how the horse moves. So this has a really long tradition dating back at least 200 years. And yeah, it enables the caretaker or a vet to detect these orthopedic problems, which problems with the legs and joints. So since a horse is heavy with quite thin legs, I mean, they are optimized to run fast. This is an important cause of injury and pain, that they have problems with the joints. So traditionally, what you do is that you have a veterinarian looking at the horse running back and forth. And the veterinarian watches the horse, but also listens to the sort of irregularities in the sound when the hooves hit the ground. And so basically the lameness comes from that the horse tries to take weight off the leg with the pain in it. So, and this takes very, very long experience to do such examinations. So the veterinarians I collaborate with at the Swedish University of Agricultural Sciences, they have in recent years integrated more and more data-driven and measurement-driven methodology, both in their research, but also in the clinical practice in the veterinary hospital connected to the university. So they do all sorts of recordings on the horse's mocap, for example, to detect irregularities. And traditionally, you, as I described in the last slide, you do this by manual inspection. And you have sort of a more holistic evaluation of the horse rather than measuring very precise metrics. So the key finding relevant for the first collaboration I would talk about is that it's good to measure objectively biometrical information using computers rather than manually. So in a large clinic like the one of my colleagues, you have the resources and space for an optical mocap system, but and this is really good, but it's of course expensive. But there are also options for more in the wild that you can have yourself in your cell phone, and they are based on video measurements or IMUs. And they are, of course, much more accessible to everyone, but they are not as exact and they measure very sparse things. So, what we thought is that let's develop something that is the best of both worlds, which gives, that is applicable in the wild, outside a very sort of resource-rich hospital, but also gives very rich measurements. So we are developing H-small, which is a 3D pose and shape model, which is extracted from raw video. So this is how it works. So we have the H-small model that we fit to a video and obtain a 3D reconstruction of the horse in the video, and so which captures as much information as motion capture, and it also captures even more information. So potentially it will display a lot more things than motion capture, like you can assess the weight of the horse, the sort of how the muscles are distributed and so on. So this work is performed by my PhD student, Celie. So I won't go into detail about this model here, but you can read more about it in this paper from this workshop last year. So H-small is a horse-specific model, version of small, developed by Sylvia Zuffi. Without the first name here, but I will change that. Which in turn is an animal counterpart of simple, which is a widely used shape and pose model for humans. So while so the bottleneck so when we're developing H-small, the greatest bottleneck is data. So it's data of the shapes of horses and how they vary. So simple is trained with tens of thousands of scans of human bodies, while the animal counterpart small is trained with only a few, so orders of magnitude less data, so only a few scans of animal toys, actually. So it's not even real animals, because it's kind of difficult to scan hippopotamus and so on. And H-small is built in the same way. It's so we have more variations of horse shapes, but it's still toys. So of course, to do is to collect more of these shapes so that we have an idea of the modes of variation in horse shape over different individuals. So this is an important focus in the future development. So how good is this? So I said before that, we want to develop this model that can replace motion capture and give biometrically interesting information. So we tested this by fitting an H-small. So we have a data set from a lab in Switzerland. So I refer to the paper for more details, where they have recorded horses. They have mocap markers on, so we have mocap ground truth. And there is also video of the horse from the front and from the side. And they run on a treadmill. So it's really, it's a very controlled scenario, but it still gives a very good view of the horse moving. And so there is labeled sequences. There are sequences in this data labeled with the kind of lameness that the horse has while it's running. So it's collected in an ethically controlled manner, more described in the paper. So what we wanted to test here was whether H-small captured all the relevant information from the motion capture. When we fitted H-small to the motion capture, did we lose anything that would be important when we wanted to distinguish between lameness and not lameness? And it turned out that we didn't, because so we classify directly from motion capture using a graph convolutional neural network, a dynamic graph convolutional neural network, and it actually, so classification from H-small fitted to the mocap actually outperforms very, very slightly, but it at least is at par with the classification from the original mocap. And even so, subject 1 was a bit problematic. I'm not sure why, but basically classification from mocap was really good, while classification from H-small failed miserably. So if we take that away, the trend becomes even clearer that it seems that H-small actually extracts even more information from the mocap. But it's just an hypothesis, so we had to verify it. But at least it captures all the important, it retains all the important biometrical information. And we were happy about that. And then, of course, this is applicable in unconstrained scenarios without markers. So you can move this outside of the lab. And next, I'm going to tell you about our current work on regression using the H-small model. So I have another MyBridge sequence here because they are so cool. We could have a completely other talk about how MyBridge, I saw Pascal Foix also mention MyBridge, how he collected these sequences. It's amazing. Yes, as I described a few slides ago, this is what veterinarians do now. I mean, so they listen to the sound as well as look at the horse. So our hypothesis here is that audio helps to estimate a more accurate pose. Because the hoof on the ground sound is a really strong cue for timing of the foot on the ground, but also the force by which so you could potentially also use, if you had a dynamic model with a spring mass model, then you could potentially also use the force information to do the reconstruction. Yes, so I refer to, so we have paper in a workshop yesterday at CVPR, and I refer to this paper for the details. But the gist of our work is that we have a fusion regression block with a neural network that fuses video and audio. So it's two separate streams, but they interact a lot inside this regression block. And out come two 3D pose sequences, which are using both video and audio. So they are two, but they are very similar because they have interacted so much. And the cool thing about this is that you, at test time, you only have to feed in one of the modalities. You can use it with just a single modality because the regression block has learned sort of correlations, basically typical poses, video appearances connected to a certain sound and vice versa. So it has learned how to correlations between the two modalities. And we also have a separate block where we estimate the shape of the horse, which of course is very hard to hear from the audio modality. And we also estimate the camera parameters. Yeah, so it should be noted that here we have no, before what I showed you was an experiment with where we fitted the model to mocap, but here we have no 3D supervision, only the 2D silhouette and 2D key points and the audio stream. So this is what it looks like. So what we have over here, so this is results for, so in the training set, we had only brown horses, and then we tested with yet another brown horse. So it's a quite easy problem. The horse is centered, and the network has learned fine how to classify. Now it stops for some weird reason. But basically, it's quite an easy case for both audio and video stream alone. And you can note that since the horse is centered in the sequence, the audio stream can also, it uses just the sound of the hoofs, but it still works fairly nicely. And fusion doesn't add a lot. But if we move to a horse with unknown color, a white horse, the video modality is completely useless because the network has no idea what this is. It has just seen brown horses. So you can see here, here the video, using the video stream alone makes it impossible to do anything. Now it stops. Let me start again. While the audio does fine, but the horse is of course in the wrong place because the audio has no idea of spatial position. But when you fuse it, it takes the best of the audio, it takes the best information from the audio and from the video and fuse them. All right. Yes, so yeah, so stop me if there are any questions, but I'm moving on. We can have questions afterwards. So automated motion analysis, as we talked about earlier, it says a lot about orthopedic problems, but horses being herd animals that communicate to others, they convey a lot of information through their facial and bodily behavior, just like humans. So we can use that as well. So back to my collaborators. So they have in recent years done some quite novel research where they have recorded and analyzed video of horse facial expressions, which were not well understood up until 2015, actually. So key finding is that horses display facial expressions of pain just like humans. So in order to investigate how horses express acute pain, so not chronic pain, but pain that happens here and now, my collaborator Pia Haubro Andersen and colleagues did a controlled data recording where they, so I should say, where they basically recorded horses which they knew were in pain or were not in pain. And they focused on the face and front half of the body. So this might seem mean, but you should remember that it's of great benefit to millions of horses if we could get a better way of detecting signs of weak pain. So we could save tons of horse lives. And this is the same type of pain induction used in humans. So it's completely reversible and you can stop the pain at any time. And so on. So there was a data set and they used it to develop facial grimace scales for objective manual assessment, which was a great step forward. But we took one step further and wanted to do automatic recognition. So we used a deep learning approach. So this work is performed by my PhD student, Sofia Bromé. And so we have a paper in CVPR 2019 where we propose a two-stream CLSTM approach to detecting pain. Yes. Yeah, so the input is, so here we took a step further also methodologically. So we do not use any manually defined scales. We just we just show raw video to the network and then a label. So we basically, so we have a two-stream network. We will have RGB video and optical flow. And there are 5-second clips of showing the horse. And there is one label for the clip, which is pain or not pain. And no additional information. And so the results show a lot of different things. So we did a baseline, veterinary baseline study where we let human veterinarians look at these 5-second clips and provide a label. And it was extremely difficult for them. So it should be noted that this is not the way you sort of diagnose a horse. You don't look at a 5-second clip of the horse. You sort of interact with it and touch it and so on and see how it reacts to different things. You sort of poke it and see how it reacts. So this is a very unnatural situation for a veterinarian, just to all the veterinarians out there. This is not how bad veterinarians do when they diagnose pain, but it shows how difficult this problem is. So it's not, as opposed to the quite, so you usually when you have these sort of classification problems, you have quite clear classes. It might be fine-grained, many classes, but they are quite clear. But here it's just a binary classification problem, but there are very subtle signs or evidence, basically. So this is a very difficult problem. And then we tried a number of different networks. So firstly, we wanted to test how important is temporal information? Turned out to be really, really important. So we have 3 networks. One that only diagnose and also so that only uses single frames, so it doesn't use temporal information. Is this one? And then we have a network that treats every frame using a CNN, and then and sort of extracts a representation of that frame, and then has a LSTM on top. And that sort of includes temporal information, but an even more sort of integrated use of temporal information is achieved by the network I showed in the last slide, which is a CLSTM, two-stream CLSTM. So we can see that the single frame model performs the worst, and then this is slightly better, and the fully recurrent performs the best. So it seems that temporal modeling is crucial. And we can also see that only the CLSTM model does well with only optical flow, which also tells us that the other models are less good at making use of the optical flow. And we can also see that the CLSTM model is able to improve with, so here is only RGB, and this is having both both streams. I shouldn't say modalities, but 2 streams, and it's able to make use of the optical flow more, while the others, they basically don't improve with optical flow. And another thing we can note is that, so as you understood from the data description, this is connected to ethical problems. The data collection is, you sort of have to put horses in pain in order to record them. And this is, of course, says that we should use models that need as little data as possible. So so one way to go is to have a semi-supervised model that makes use of unlabeled data, because we can, of course, record a lot of horses that might or might not be in pain. I mean, after surgery, for example. But another way, sort of no-brainer is to use a model with fewer parameters. So the CLSTM has only 700,000 parameters, while the other networks have several million parameters. All right, so the next thing, so it's all good. So we found that the CLSTM performs a little better, but it's also interesting to look at what they actually learned, the different networks. So so here are 2 sequences, one which is no pain and the other is pain. And we see here on the top, we see the CLSTM GradCam visualizations, which basically are, I'm sure a lot of you are well aware of GradCam, but shortly, GradCam provides a heat map with areas of importance when making the decision. So it's basically, where does the network attend when it's making its pain, no pain decision. So if we see at the CLSTM, firstly, it focuses on sort of the same point. It's sort of consistent over time. It looks at here, it looks at the muscle or the nose of the horse, while the network that first processed each frame and then had a LSTM, it sort of jumps around. It looks up in the corner, on the ear, and then it looks in the corner, it looks on the neck. Yeah, it looks at many different places. And the same thing here. Here it looks sort of at the neck of the horse and at the eye, which is interesting because the eye is really important for facial expression. While this one jumps around. So it gives sort of indication that the CLSTM model sort of attends to the right things. But of course, we need further exploration. So in the future, I mean, if we develop, if we have these kind of models that we train with much more data, much more data from a lot more situations so that it becomes really knowledgeable, then you could even learn things by inspecting the model. So firstly, you could, of course, develop applications such as a decision support system for veterinarians that they could have in their iPhone. And of course, it wouldn't replace veterinarians, but it would assist them giving additional information. But potentially, you could even learn new veterinary knowledge from these models. So you could basically inform, you could improve veterinary training, even if you're very sort of visionary, you could, so you could learn what to look for. And basically, veterinarians are, they train by doing. So they learn how to get a sort of clinical eye or gut feeling about if a horse is feeling well or unwell. And it would be interesting to use these methods to sort of inspect this implicit knowledge and make it explicit so that you can train other veterinarians. All right, so back to our method development. So our method from 2019, it was able to recognize pain, but it was a setting which was quite sort of artificial. It was very short sequences where the horse had clear pain behavior all the time if they were in pain. But it would be interesting to be able to detect more realistic types of pain. So in the next paper, which you can have a look at in the poster session, so you should come to poster number 3 and talk to Sophia, who will be there. I will be there as well. And learn more about this work. But otherwise, you could read the +1 paper. Yes, so now I'm running a bit out of time, so therefore I will refer you to Sophia's poster, but I will shortly describe what we've done. So we had the same data set as before with these clear pain signals. And then we also have a new data set collected by another colleague, Katrina. And it contains a different kind of pain, which is much more similar to the pain horses would experience in the wild. And we compare 2 different models. And what we could see is that both models achieved really good results, as we saw before on this data set that we had before. But when we moved to this new data set, they were basically not better than chance. Yeah, so it basically doesn't work very well. And then we can, then we have tried different kinds of fine tuning, and what we found was that even though the 3D CNN based method performed better in the original domain, it the COM-LSTM model, this recurrent model, it was able to generalize better to a new domain, which was interesting in itself. And there is a great variation between the different horse subject, and it didn't help with fine-tuning really. And we also did a human expert baseline comparison, which was really interesting, which I will refer you to Sophia's talk. Basically, we found that, again, that our COM-LSTM model greatly outperforms humans on these tasks, which says that it's really beneficial to develop these kind of decision support tools, because they will of course not be as good as a human veterinarian in weighing in all the information about the horse and making a diagnosis, but they could give a lot of evidence, that precise information to the human expert, which could be valuable. So come and see Sophia's poster. Yes, so back to our initial statement that we need to detect pain in order to increase animal welfare. So horses are herd animals with a really sophisticated inner life. So you could think about having an animal version of the Maslow hierarchy of needs. I don't know if it exists. I would be very happy with feedback. But basically, I think that we should, horse welfare includes ensuring that horses are stimulated, interested in their training, getting enough sleep, and so on. So we should also look at other things than just the absence of suffering, basically. And you can use the Mendel valence arousal scale to sort of categorize different emotions in horses. Yes, to conclude, so we do a lot of research on detecting automatically signs of pain in animals. And in the future, we would like to continue this, and even maybe veterinarians can even learn from these deep learning models, and we can recognize other expressions. Thank you so much.
Thank you, Helvig. It was a great talk. It's very inspirational, and so we have some time for a couple of questions. I'd like to start with one in the Zoom. Sylvia Zulfi asked a question that, do you think the pain expression cue could be shared among different species? And maybe it could be exploited for using more data.
Yes, absolutely. So I actually had a slide about this, which I removed. So I think that horse is a very good model animal to work with, because there are many similarities in, for example, facial expression between horses and other pet animals, such as dogs, cats, that have developed together with humans and are sort of developed to communicate with humans. There are these facts, so equifax systems for expressing facial expression, and there is also dog facts and cat facts and so on. There is also a lot of similarity in both in how like body behavior and also facial expression to other herd animals, such as cows, sheep, and so on. And why I think it's good to focus on horses rather than, so why I focus on horses rather than, for example, a wild animal, is that there is a lot of data from horses. Horses are recorded a lot, and a lot of money is spent on a lot of resources are spent on horses. But definitely it would be super interesting to see the same kind of research in dogs, cats, cows, sheep, pigs. Yeah.
Thank you. So we have another question.
And did you ever see? Thanks for the talk. It was great. And a question about the H small versus motion capture comparison when classifying lameness. First part. Have you looked into how much the shape information is being used for that classification versus just the pose?
No, we have not. So in that experiment, we didn't use the shape information at all, actually, for classification. But
you just got the pose out.
I got the pose out of the H small model.
Okay.
Yeah.
Do you think that there's a lot in the shape channel that could be useful? What's your
not for lameness, but definitely for assessing body mass index, for example. So like health status, muscle volume, things like that, definitely. And the shape also helps the fitting, since you explain more in the image. It sort of makes the fitting more robust. So if you have many animals, for example, occluding each other, the shape estimation will help make it more robust. So I think in the end, it helps the tracking.
Oh, thank you.
All right, so I think we are time's up now. Helvig, thank you, and let's thank speakers. Next speaker is Amir Patel. Amir, can you share your screen, please? Amir? Yeah. So can you hear us? Hello?
Hello, can you hear me?
Yeah, we can hear you.
Sorry,
Right, great.
I think I'm muted by mistake. Okay, so can you guys
So
see
let
my
me,
slides?
Amir, let me introduce you first.
Cool.
Okay, so I am excited to introduce Amir Patel, who is an associate professor at the Department of Electrical Engineering in University of Cape Town, South Africa, and director of the African Robotics Unit. And Professor Patel spans a diverse range of computer vision and robotics problems. Especially he's interested in the locomotion of humans and animals that can inspire robotics, manipulation, and locomotion. And what makes his work so exciting is that he's really working with real wild animal living in a natural habitats, such as cheetahs. And so he's a recipient of numerous awards, including Claudette Leon Emerging Researcher Award, Oppenheimer Memorial Trust Fellowship, and Google Research Scholars. So Patel, is all yours.
Okay, thank you very much. Yeah, thanks for the great
Patel,
yeah, can you hear me?
We see there's some black box on top of your slide.
Yeah.
That's one, seems like a window pop-up.
Is there a way that I move that? I can move it down. Is it this one?
Can you minimize that zoom bar? I think you can minimize.
Oh, okay. Do you know how? Because I don't know how. You're using doc? No.
Yeah, bottom is better.
Bottom is better. Okay.
Yeah.
Better? There must be a way I can hide this.
Yeah, it's better. Okay, please go ahead.
Okay, I think I'll just start. So yeah, thanks for the great introduction. So today I think I'm going to talk about well, and what I would like to do is sort of give you guys some context into where I'm coming from and the work that we've been doing in my lab for, I guess, almost 10 years now. And our main sort of goal is to understand cheetah locomotion. And as you all know, and I've seen lots of talk today on on animals and how much more tricky they are to work with than humans. So my talk is titled Biomechanics Unleashed Towards an Understanding of Cheetah Locomotion in the Wild. Okay. So my journey really started from trying to understand maneuverability, and things like jumping, acceleration, sudden stops or turning are things that are really going to be critical for future robotic systems if they are ever to leave the safety and the confines of the laboratory. So if you want to make our systems more autonomous, they need to be more agile. And maneuverability is quite interesting because it's a really nice case study for studying competing requirements and how animals have negotiated things like compromising on safety, economy, robustness, and agility. So weighing up all of those things to get the best solution. But it's quite interesting because maneuverability is still sparsely studied compared to steady state locomotion. So for steady state, constant velocity motion, we have we even have a vocabulary for things like gait. We have things like trotting in quadrupeds or walking or bounding for quadrupeds as well. And what's also quite interesting is that animals are really extremely adept at doing these things. They do them with ease because they are sort of like paramount to their survival. And no animal epitomizes maneuverability like the cheetah. So this is just some examples where you can see, the extreme agility of the animal. Also, you can see the things like sliding, turning, using its whole body, to maximize its ability to maneuver. So, what do we know about the cheetah? So the cheetah is the fastest terrestrial animal. I think we all know that. It's actually been measured to run up to 29 meters per second. And what's interesting is that some work by Alan Wilson's group has shown that the hunting success isn't actually about them, running at the top speed all the time. I think the maximum speed they got was 25 meters per second in their paper, but it's actually more the ability to rapidly turn and rapidly decelerate in response to the prey's maneuvers. So another thing that we know are these sort of like facts, which are, I guess, more anecdotes where people say things like the cheetah tail is considered to be high inertia. It's also considered to be heavy and used as a rudder or stabilizer. If you watch any any wildlife documentary, if you're in South Africa and you on a game drive, you'll hear some a ranger will say things like that. But there's actually no one's proven that at all or done any kind of biomechanical study on that. So when I started my PhD way back in 2012, I was really sort of excited about doing bio-inspired robotics, and I thought we have lots of cheetahs here in South Africa. What I'll do is we'll observe biology, and then my goal was to build the next generation highly agile robot, and I would do this by leveraging off what I had thought would be, a wealth of biomechanics literature, things like lots of data and models, but to my dismay, none of those things actually, existed at the time. Sorry, I really need to move this. Oh man, I can't. Maybe I can. Oh, okay, I can move it to the next screen. Is that better?
Yeah.
Okay, awesome. Thanks. Okay. So yeah, unfortunately, like this data was non-existent. There was like very little that we knew about the cheetah's locomotion. So what I thought was, okay, could I use my knowledge of robotics to provide insight to biology through things like new models, both simulation and physical models in the form of robots, and then also provide new ways of collecting data. And that's sort of what we do in my lab. We study the cheetah as a model animal for maneuverability and rapid locomotion, and we employ methods which we are actually roboticists, and we are trying to understand biology through the lens of the techniques that we know, things like multi-body modeling, feedback control, trajectory optimization, and optimal control. We use robots, physical robots, as experimental platforms, and then lots of state estimation and computer vision, which is, I guess, sort of why I'm talking in this session. So yeah, we do lots of those kinds of things. And in my PhD, what I did was there was no kinematic data or biomechanical data, on the skeleton or the tail even of the cheetah. So what I did was I developed various robotic platforms based on sort of the motion primitives that were observed in wildlife documentaries. So I developed this robot called Dima, and it's comes from a Sotho word, which is a South African language, and it means flash of lightning. And what we discovered is the motions that we saw in the roll axis, on the left, they are able to increase a robot car's ability to rapidly accelerate. And then also the motions we saw on the pitch axis are able to, increase the ability of the car to rapidly decelerate or accelerate. You can see the videos. And then we also saw something interesting in the videos where, the cheetah sort of swung its tail in sort of a cone fashion. And we found that that's the optimal way of swinging your tail when you're trying to produce longer duration stabilizing torques in the body. But what about the cheetah's tail morphology? So as I said before, it's often considered a counterweight. But in 2014, I was invited to a necropsy at the National Zoological Gardens here in South Africa. And to my surprise and little bit of terror, to be honest, it was only 2% of the body mass. So I built all these robots beforehand, and show that the tail makes difference and so on. And I thought maybe we had some weird cheetah, but we measured this on 8 cheetahs, and it's actually only 2%, and the muscle mass is really concentrated quite far down the base of the tail. If anybody's interested, I can show you some very gruesome pics. What I did see though, which sort of piqued my interest, was that if you can see that picture, the tail is the volume of the tail is quite large, but when you remove the fur and the skin, it's really, really skinny. So like any good engineer, I thought maybe there's aerodynamics involved. So we took our cheetah tail and our pelt, and we actually put it in a wind tunnel. And after various experiments and some modeling, we discovered that the aerodynamic effect actually makes up 40% of the angular impulse exerted on the body. And this is, I guess, it was surprising, but also not so surprising in a way, because everything about the cheetah has been optimized for speed, reduction of weight, reduction of, the size of the skull to reduce drag. And why would it need to carry around this heavy tail, whereas it could use sort of like, if you imagine you're putting your arm out of the window in a car as you're driving, you can feel that massive amount of force. If you're moving fast enough, you get a quadratic effect on the torque. So what we saw is that the effective tail area, so what the tail behaved like, is something that looks like double the area, but it can create more force without the mass penalty, right? So it's all just sort of like fur, which isn't really heavy. And we tested this idea further during my sabbatical at Carnegie Mellon in Aaron Johnson's lab, and we actually tested it on the Minotaur robot, and you can see we use an extremely lightweight tail, and we were able to do self-writing with the robot we launched it in an unfavorable position, and it could land nicely on its feet. So this was pretty cool. But the most important bit has always been, what do real cheetahs do? And it's something that's bugged me for a long time, after my submitting my PhD, and criticism I had of, I guess, of the work was, we've shown that robots can benefit from these maneuvers. Our simulated models, the fur makes a difference, but like, what did the cheetahs do? So in a perfect scenario, if you're trying to understand the biomechanics of something, we put it in a biomechanics lab, you put it in front of a Vicon camera system, and you'd, get the subject to walk on an instrumented treadmill, giving you 3D force and center of pressure. And, you could really go to town and do inverse dynamics and all kinds of nice things. But obviously we're dealing with a wild animal here, so it's not really possible. So skeletal motion capture, how do people do motion capture on wild animals? Most of the time, the majority of people are using IMU and GPS collars. But this is actually only telling, part of the story. So if you think about it, you've got this animal which is running, turning and doing all kinds of amazing things. And a GPS collar and collapses that down to, a single rigid axis. So a single lumped rigid body, and you've lost all the articulation of the legs and the spine and the tail and even the head, which I'll talk about a bit later. And so obviously an IMU GPS collar isn't going to do the job if you're trying to really understand how animals are able to maneuver. So I developed a system, and I thought, if I can't get the lab, I can't get the cheetah to come to the lab, I'll get the lab to go to the cheetah. And what we did was we designed a harness system which consisted of an IMU and GPS, 2 stereo cameras which faced backwards, because I was quite interested in understanding the spine and the tail, and then some wireless IMUs. And then we could fuse all that information to produce skeletal motion of the cheetah, sort of in a free running setting. So we designed a system actually based on a smartphone, and smartphones are great. They have IMUs, GPS. This one actually has a barometric pressure sensor. We have some wireless IMUs, which we put on the tail. And then we also had 2 GoPros running at 100 frames per second, facing the back of the cheetah. We then did some state estimation. So I'm a big fan of the Kalman filter, and because this was an offline problem, we could do Kalman smoother, and we fused that with a skeletal model shown on the right there. So 2 links on the spine, and then another 2 links in the tail. I won't go into details of the Euler angles and things like that. But using that information, we can then, produce the skeletal motion. So what does that look like? And this is not sped up or anything. This is real time. Okay, so that's sort of what it looks like riding on the back of a cheetah. And unfortunately, the system worked great, but was too invasive for most of the cheetahs. We only had really 2 or 3 that were okay with running with the system. So what I then did is I approached GoPro, and they sent me 12 nice cameras and some of the 2 of the breeding centers in South Africa. And what we did was we placed up 2 sort of camera arenas in each of those centers. And what the cheetahs do sort of daily for enrichment is they chase a lure around a track. So it's usually either like a piece of rabbit pelt or plastic, and they chase it around the track really fast, and that sort of gets their prey drive going and gets them exercising and so on. So what does that look like? We've got some really nice data. You can see over there the handlers trying to get the cheetahs to run. So these are captive bred cheetahs. But this is a really, really challenging data set. So as you can see from the video, like the different times of the day, different seasons, because we sort of collected this over a period of 3 years. We had different cheetahs. You see, one of them was even a subvariant of the cheetah called the king cheetah. Angles, distances, occlusion is a terrible problem. And then the worst problem of all is that not all of the cheetahs had markers on them. So I think one in the video had some pink stickers we were allowed to place on them, but most of the cheetahs didn't have any markers. And that really posed a challenge for me. I went on my sabbatical in 2018 with this data set, thinking I was going to write a nature paper on cheetah tails. But yeah, we really struggled for quite a while to try and reconstruct the motion without markers. And just so you can get an idea of the challenge of the data set here, you can see the bounding boxes. The cheetahs really vary, massively in scale because we have no control of where they're going to run far or close in front of the cameras. But then I was very fortunate to meet Alexander Mackenzie Mathis at Harvard at the time and now at EPFL, really smart people. And we worked on co-authored a paper which was featured on the cover of Nature Protocols, which extended the deep lab cut system to, actually applying it to cheetahs in the wild. So how does it work? We give it the 6 views of the cheetah. So you can see there's an example. And then DLC, I'm sure most of you know, deep lab cut gives us the 2D pose estimates of the cheetah skeleton. And then we can do some nice, 3D reconstruction, or so I thought. So there was still some problems. You can see here, there's a massive problem with outliers because the data set is so varied. You can see different angles. Look at the sun, really just, saturating the image. It's also quite a bit of noise. You can see there. Really, really challenging data set. So what we did was we extended this work and developed a new algorithm called full trajectory estimation, which we presented at ICRA last year. And how it works is we take 6 DLC labeled camera views. We calculate the noise covariances for each of them. We then take a model, so a kinematic model and constraints for the cheetah, and then we can reconstruct the 3D pose, and we can run this optimization on the entire trajectory simultaneously because we aren't, this is in real time, and it's quite great because it handles all the constraints. Because I tried using a Kalman filter, and you can see in the paper, and sometimes you get weird poses, the spine's bending the wrong way or the arms bending in a weird fashion. So it's really nice in that way. So this is how it works. You can see here the 6 views, and here you can see the cheetah running. And this is a comparison of the 3 methods we did in the paper. So just a straight up SPA triangulation, and then an EKF, and then our new trajectory estimation method. So yeah, it looks pretty cool. You can see we can even do maneuvers there, like things like diving with lots of occlusion and sliding and things like that. You can see dust is a very big problem in this data set. Okay, so as part of the paper, we also released the CINO set, which is a new data set which contains 10 different cheetahs, 120,000 multi-view frames, 7,500 hand annotated frames, and then we've also got the 3 reconstruction baseline. So we definitely check it out if you want a challenging, data set to try out. And yeah, I think it would be great if more people like sort of applied their methods to it. So yeah, so what can we do with this data? So the first thing I did when I was watching tons and tons of these videos is I noticed that the cheetah's head remains really, really still. So you can watch us chasing the lure, but the head is sort of locked onto the target, despite the massive amounts of body movement. And then I thought of, is there some kind of head stabilization happening? And, with a very simple model we can construct here, you can see that there's way more deviation in body angle versus the head pitch angle. And you can, just an example trajectory, you see lots of up and down body pitch, but the head angles being sort of smoothed out. We do know that the cheetah's inner ear is specialized for measuring really high bandwidth rotations. So could there be some high bandwidth control at work? And that's what we're currently looking at. And then the tail work, which is what I'm really interested in. We did a very simple two-body model here with a virtual tail and a virtual body, and started looking at acceleration of the cheetah versus the tail's angular velocity. It's not quite as strong a correlation as I would have liked, but I guess it's an initial start. I think we need lots more data. So how can we improve things? So I guess the first thing is we need more data. And you might think, well, you have tons of data, but many of the videos in AsinoSet can't be reconstructed. And you can see on the side this because many of the times, because this is a wild animal, it's doing all the cool stuff in the corner of the frame. And maybe there's only one camera seeing the cheetah or one side of it, or it's really small. So we can't really do any reconstruction. So I thought, what if we moved the camera to the cheetah? So this is what we did in our recent paper. My students Aman Valley and Daniel Joska developed this automatic tracking 3D pearl system. So how it works is we take a frame, we have a webcam, and we use YOLO V4 Tiny. We find the bounding box. We then have an extended Kalman filter, which gives us a prediction of where the cheetah bounding box is going to be. And then we have a full state feedback controller, sort of turning the motor and the camera, the stereo camera set up towards the cheetah. So this stuff is under review at IROS. So you can see this is an example first on a human. That's my PhD student Naoya running there. And you can see we can reconstruct in the world frame despite the cameras moving. And there's a nice video of the cheetah. You can see moving across frame. And we're now able to reconstruct while the camera is moving. Okay, so that was more data. What about different data? So we can capture quite well, I think, what the cheetah is doing, but we still don't know how it does it. And this is and a way to do that is to get force plate data. So we collaborated with Alan Wilson, who's at the Royal Veterinary College, and he had his former PhD student had tons of force plate and high-speed camera video footage. And using this information, we combined it with our trajectory estimation as well as with deep lab cut. We could then estimate the torques and the forces exerted on the cheetah. So I won't go into detail about this too much, but my student Zico is in poster session is 2, number 26, I think. You should go and check out his talk. So just a sneak peek of what it looks like. So here we see the cheetah and it runs on the force plate, and now we can actually predict the torques of the cheetah as when they're running on the plate. Okay, so the next one is one problem I had and criticism is that the captive animals often don't have the same motivations. So often, as you see at the beginning of that video, the cheetahs don't want to run, so they need to sort of like egg them on because they're going to be fed anyway. And wild animals are way more active because they actually need to hunt to survive. So how do we do remote whole body skeletal estimation in the wild on actual wild animals? And this is a sort of crazy idea I came up with where we'd use a high-speed camera with a telephoto lens and a LiDAR, and then fuse all that information to give us 3D skeletal information of wildlife. And I want to show you sort of how far we've come on that. So we've done a mechatronic design on that. So here's the CAD model. This is us doing some testing as we speak, and I'll show you some results. So this is on game ranges pickup, and you can see that it's in the window. And here's the system over here. It consists of a LiveVox Tele 15 solid state LiDAR We've got zoom and focusing motors for the encoders. We've got pan and tilt rotary encoders, so we can get the exact angle. Touch screen, joystick, a Zimia high-speed camera with a telephoto lens as well. So this is some initial data we took at Anne van Dyck Center. So you can see it on the right is the high-speed video, and on the left is the LiDAR point cloud. And you can see, if you look, you can clearly see the cheetah coming towards us now and coming down here. You can see doing a nice cool flick and turn. Okay, so then we can take that information and we can fuse it. So the camera is an example, and we can now do 3D reconstruction of this animal, and it's 100 meters away. So that's pretty cool. So I wanted to share some very, very sort of hot off the presses results. So we were in the Kalahari Transfrontier National Park here in South Africa. I was there until Friday, and we collected this footage I'm going to show you on Wednesday. So we were very fortunate to catch a cheetah, and so a cheetah mother and her 3 cubs hunting and killing a springbok. And you can see here, this is what this looks like. So you can see the mother taking it down, and this is about 70 meters away. So what's also quite cool is you can see on the right there, you can clearly make that out in the point cloud. So now we've also got depth information, from which we can correspond through an extrinsic calibration to the camera data. So we haven't reconstructed this yet. I pushed my students to do this, but they're still out in the field busy collecting this data. So in conclusion, studying cheetah locomotion is really challenging, but the understanding can help leg robotics as well as biomechanics. So, we can understand, how animals, sort of control these rapid maneuvers, but the data is really on a critical path for more insight. And it remains a challenge, I think, for a computer vision and machine learning community, for us to understand, wildlife locomotion in natural environments. I think it's going to be really key. So some references, thanks to all my nice collaborators and sponsors. I wanted to send out the last plug is I'm looking for a postdoc in multi-sensor fusion for animal biomechanics. So University of Cape Town, with the top ranked university in Africa. Cape Town is a lovely city for anyone who's ever been here, and you get to work with cool animals. So if you're interested, please pop me an email, and yeah, I'd love to chat. So yeah, thank you for your time. Are there any questions?
Yeah, it was a very inspiring talk. So any questions from audience first?
Hi, Amir. Super cool work. It's really exciting to see the progress that you're making. I'm kind of curious about the last work where you're using the LiDAR to fuse the information. Like that is far away, and so it requires your students to track them, or like how do you maneuver the camera? And
So
like,
how do we track the cheetah?
or like, yeah, if it's far away and then there's like a telephoto lens, like so your students are like basically filming the cheetah.
Yeah, so it's very difficult. It's very difficult. So I mean, there's some photos I didn't put up of myself actually, like also like trying to do it, and it's very tricky because it's very far, and the cheetah is so fast. So we missed some footage in the beginning where I was actually for this video I was capturing it, and we were on the springbok, and the springbok ran across. And I was locked onto the springbok, and then out of nowhere the cheetah came, and once it grabbed the springbok, we sort of like had to catch up. So it's very, very tricky. I think what we should do in future is to actually get a professional camera person out there, because I thought we could just do it myself and my PhD students, but it's very, very difficult. Another challenge is actually finding the animals. So I think so. This was our first trip, and it's quite cold there, even though it's the Kalahari desert, but it gets down to -2, well, Celsius, I should say. And we really haven't seen many cheetahs in the week we've been there. We've only seen, I think, 2 days of actual cheetahs. We've seen lions and leopards and giraffes and all kinds of animals like that. But no cheetah, not many cheetahs, sorry. I hope that answered your question, Angju.
Yeah. Super cool work, and we're always looking forward to your chapters so that we want to reconstruct them.
Thanks.
Thanks a lot.
Hi, do you hear me?
Yeah, go ahead.
Okay, so my question is related to the Andrew's question. So when you use a LiDAR data, so is it possible to find the cheetah automatically from the LiDAR point cloud, or was it done by manual?
So is it possible to find the cheetah directly from the LiDAR? Is that the
Yeah,
question?
from the LiDAR point cloud. So did
Oh,
you
yeah,
find the
yeah.
cheetah by hand, or was it by some kind of a detection method?
Good question. Yeah, so, I mean, I didn't put as much detail in there as I'd hoped. So how it works is we've done a really nice calibration, extrinsic calibration with a large checkerboard. So we know the camera pose. Sorry, we know the LiDAR pose relative to the camera. So what we do is we use deep lab cut to find the joints of the cheetah, which we can do very well now. And we then reproject those into the point cloud. And then the point cloud is still quite sparse, if you can see on that video. So what we do is we find the closest, the brightest, closest pixel. And that gives us the depth. And what's also challenging is that, so the video footage is coming in at 100 frames per second. So we've got, 2D pose predictions at every 100 hertz, but the LiDAR only comes in at 10 hertz. So we only get a depth estimated at 10 hertz. What we're going to do in future is to train the neural network to simultaneously take in camera and LiDAR data, and then give us 3D pose. Yeah. But it's obviously that's a challenge because we don't have enough training data.
By the way, so how large the checkerboard is? So you say that you prepared a large checkerboard. So
Yeah.
How
large is it?
Yeah, 3 meters.
Oh yeah, it's about I would say 1.5 meters by 0.5 meters. So yeah, we have some very strong grad students and they lift it up and move it around.
Thanks so much. Great work.
Yeah.
Cool, man.
So I have one quick question. So you started your talk with the biology versus robotics, where you want to get the inspiration from biology for robotics on the other way around as well. I just wonder whether you have learned something from this biology, so from the motion capture that can help for the robotics, or the other way around.
Great question. Yeah, so I think what's surprising with this is that how much work we've had to do in computer vision and state estimation, and we've also done, I didn't show, but like a whole host of optimal control and trajectory optimization, novel work that we've had to do, to study this animal. But what's also interesting is what the animals told us. So one thing that's quite apparent is that stabilizing your sensor matters. And what I mean by that is, we've got lots of quadruped and bipedal robots now, and the main goal of the robot is, to actually collect data. But most often they are just mounted straight onto the body of the robot. And if you look at the cheetah, despite it running and bouncing up and down, the head, which is the main, sensor, is stabilized very well. So I think we need to look at stabilizing our, our LiDARs and our, whatever sensors we have on our robots. I think that's important. And the second thing is, I think aerodynamics does matter. So I think I always used to laugh in first year physics, you always ignore aerodynamic drag. But I think the animal is really exploiting it. So I think if we're going to be moving robots really fast, we need to start thinking about how we can, use the actual, environment around it to, to make it more agile. Yeah.
All right, thank you. I'm sure there are more questions, but I think we have to end here. Thank you. Thanks. Let's thanks the speaker again.
Cool. Thank you. Thanks very much for having me.
All right, so next speaker is Timothy Dunn. Are you able to share.
Did you start it?
Oh yeah. Okay. Okay. All right. So let me introduce Tim Dunn. Tim is assistant professor at Department of Biomedical Engineering at Duke. He received his bachelor degree from Berkeley and PhD from Harvard in neurobiology. And his work spans functional brain imaging, understanding behaviors of fish and mouse, and developing a tool for the neuroscience. And particularly, I found his work is so exciting because one of his work that is inspirational to computer vision, as he has shown like 3D mouse tracking using a multi-camera system. So as a computer vision researcher, I'm so excited to hear from him about, and also I like to see what he has done so far. Okay, great. Thank you. Hi, everyone. So I'd like to start by motivating the need for quantifying more natural behaviors. I think Amir just did a great job of. Maybe you can just hold it if you want. Of showing like proper natural behavior. We don't do anything near that in the lab, but it's still I think a good example of the current issues with the state of behavioral quantification in neuroscience. So the typical thing that you'll do in the lab measuring behavior is measure some low-dimensional or constrained behavior. And this is at odds with the high-dimensional neural recordings that we can make with all the new state-of-the-art tools for measuring brain activity. Right? And what we really want to measure are things like what Amir showed, right? Some sort of natural high-dimensional behavior that's actually utilizing high-dimensional state of the brain. And so I'm going to talk a little bit about my efforts to develop technologies and data sets for quantifying naturalistic behavior. So basically, laboratory behaviors are more natural than that thing on the left. Okay. And what this has done to help us study Parkinson's disease and Fragile X and some other applications in biomedicine. Okay, so I'm going to start by first people that don't know why, probably all of you know why would we be interested in measuring behavior in 3D. Wanted to understand the brain or to develop new tools for biomedicine. So 2D tracking has come a long way in animals and the lab. It can be immensely powerful for certain applications. So this is a video from 2015 in eLife where they are measuring mouse gait really precisely and using that to understand cerebellum, other diseases in mouse. The problem is if you are interested in the naturalistic behavior where the animals are sort of freely moving in an environment, you run into issues with making just two-dimensional measurements if it's not just like a top-down view on video. This is a figure from a review where you're doing flatland, and you can see the different types of ambiguities one counters if you're making two-dimensional measurements of three-dimensional freely moving animals. So one is you just get this pose ambiguity. So if you're making just from a single view a measurement of a 3D pose, there's actually multiple 3D poses and sizes of animals that are actually consistent with that measurement. Related, but slightly different, is the idea that for a given pose, right, depending on where you're making the 2D measurement, right, depending on perspective, you get changes in the relationships from those two-dimensional key points. This leads to sort of phantom kinematics, right, and dynamics that aren't actually real. Here's an example from the DeepFly 3D paper that I think really illustrates this point beautifully. You have that fly on a ball, right? You're making two-dimensional measurements from different angles of the fly. Use a behavioral mapping approach to say these are the different types of behaviors that the fly is executing. For the same videos, you're just using the 2D key point estimates. Those are here. You say that the fly is engaged in very different types of behaviors, despite it being engaged in identical behaviors, just viewing behaviors from a different angle. This is ground truth types of behaviors you get only if you measure the 3D pose in these animals, right? So this sort of motivates the need for having the ability to measure 3D and not 2D pose in these laboratory, freely moving, naturalistic environments. Is it wiggling? Okay, again, this is sort of, you all know this, but it's the state of the art for 3D pose measurement is still motion capture. So marker-based motion capture. In marker-based motion capture, you have this calibrated camera array, large set of cameras. You're tracking these little beats on an actor, very effective and highly used in human subjects. And then once you've detected these points in individual cameras, you can reconstruct a 3D skeleton. This is not ideal for animals in the lab for several reasons. One is that it's expensive to have a ton of cameras. I know Hyunsu has done this, right? But not everyone, right, in the field can create a huge, panoptic camera arena in order to make these measurements. It's hard to get animals to wear markers. So a marker-based approach is, often not feasible and can perturb the natural behavior of these animals. And, in principle, you also, need direct line of sight to these markers. And so if you want complicated arenas that are more and more natural, right, if you have these occlusions, you run into issues, right? So ideally, we have a system that looks something more like this, right, which is what people have done in the human markerless motion capture field. We have maybe a multi-view setup, but with a fewer number of cameras, synchronized video, animals moving around in like a home cage, it's slightly enriched, and then there's some algorithm, maybe it's not a neural network, there's some algorithm that then gives you the pose. So in the human markerless motion capture field, right, a lot of progress has been spurred by these amazing large data sets, right, for training these algorithms. This is one of many. Human 3.6M data set, 3.6 million frames of synchronized motion captures, 3D pose with 4 RGB cameras. You have the actors in different types of behaviors, and then you can use those to balance training data or to benchmark on different types of poses that you train your algorithm. But we don't really have this at all, or we didn't at the time for rodents in the lab. So what we first set out to do is realizing the power of having these data sets for training these algorithms. We actually acquire a data set that will enable us to train algorithms customized for laboratory rodents. So Jesse Marshall, postdoc in Bence's lab, who I was collaborating with, collected this data set, rat7m, so 7 million frames of rat motion capture, synchronized color video. So the way he did the motion capture is that he used ear piercings on 20 different parts of the rat's body, tracked those with a large motion capture camera array, and then there were 6 color cameras calibrated in the same system and synchronized with those motion capture cameras. So we were getting these 3D poses out from the motion capture together with that synchronized color. So here's a little schematic of what that looks like. So the animal down there, little markers that are attached to it. It has this little head cap on the top that had markers, which help to both affix markers to the head, but also the type of head cap one would use if you're going to do brain recordings from that animal while they're free. And then here's some examples of some of the raw data, right? We can triangulate the motion capture data and then reproject that into the color video so that you can both visualize where those markers are, but also use 2D labels for training your algorithm of choice. We also, we can't tell the rats to engage in different types of behaviors, but we can try to automatically classify their behaviors and then group those into a set of sort of coarse behavioral categories and use those as an equivalent of the action categories in human 3.6M. And we'll talk a little bit more about this mapping strategy or behavioral clustering strategy later, but suffice it to say, we can take all of the measured 3D information from the motion capture and then embed them into some space that has some organization where different types of behaviors are parts of that space. And we can label each behavior according to where it goes into the map. Behaviors are all in one type, one part of the map, and rearing behaviors in another, etc. Okay, so that was rat7m. We also recently published a new data set for social animals. So 2 rats behaving together in an arena. We gave this a different name that's not quite as catchy, the pair R24N, which is 24 million frames of 3D motion capture and color video for social rat. Similar setup as before. So we have the same motion capture system. The 2 animals are behaving in the arena. We also, because of occlusions, couldn't really track the limbs. So we actually didn't label the limbs in this data set. And in order to track the identity of the animals consistently over time in these large data sets, one of the animals always got 2 little extra markers, which allowed us to distinguish the 2 animals consistently over time. Here is an example of some of the air data set. So on the left, that's motion capture data. You can see what's kind of weird because there's no limbs being tracked. The animation is up again, the sort of embedding behavioral clustering analysis that we do. And now a live video of the animals interacting, and you can, depending on where the little point is in the map, that's the type of behavior that that animal is executing at that moment in time. And then over time, they visit different points in the map as they're engaged in different types. Little bottom scrolling thing, something we call an ethogram, which is just another sort of barcode representation of the different types of behaviors these animals are. The color video data from the data set looks something like this. This is raw data from pair with the mocap data projected into the frame to one of the 6 cameras. On the top, you can see the automatic behavioral labels that we're applying in order to group the data by category within the social data set. Okay, so we have these 2 data sets, and so we also, as Amir showed, when we just tried naive triangulation of 2D points, it was also a little bit noisy. We used a different approach to solve this, where we developed a network we called DANCE, the 3-dimensional aligned neural network for computational ecology. I'm not going to go into too many details. The idea is that you create a volumetric representation of each of the images, and then you can use that as input to a 3-dimensional convolutional neural network, and then train with the ground truth 3D data that we have from these data sets. And it does better than just a naive triangulation of independent 2D detections, which are shown there on the right. So we applied that first to single animals from the RAT7M data set, and also in other individual animal species, so in mice and birds, and in rat pups, also in marmosets. But I'm not going to show much of that data. I'm going to show you a little bit of the sort of simple baseline we have now for social tracking using that pair data set, and also some markerless data that's been labeled by members of the Ovetsky lab. So what we did in order to do social tracking using this DANCE network, so this sort of volumetric representation of these images, is that one thing we do for that DANCE model is that we essentially create a 3D bounding box in space into which that volumetric representation is embedded. So it's not a 2-dimensional bounding box in the image, but a 3-dimensional bounding box in metric space, into which we've inserted image features that we've sampled. Because we have that 3D bounding box representation, one trivial, simple way to do social tracking in the multi-animal case is that we just find both animals and insert them into their own 3D bounding boxes. And we pass those through the network as we would with a single animal. And so using just this simple baseline, oops. So these are predictions from DANCE model that we trained. We compared a few different DANCE models. The details aren't really important. Suffice it to say, at least on the pair data set, it does pretty well at tracking that limited set of key points. The limbs are much harder. Yes, question. Yeah, so that's done via the first step, which is creating the bounding box. And so the way we create the bounding box is that we just detect a single point on each animal, then triangulate that to 3D and use that to anchor the 3-dimensional box. And so all of the identification is done at the level of finding those points reliably. In this particular case, with the massive data set, we could do that without any special tricks. In the data I'll show next. So this is now fully markerless, tracking the limbs as well. In this case, in order to reliably identify them, we had to paint the animals slightly different colors so we could reliably identify them during that first step when we found the animals. So Diego and Udne, who are interested in diseases of social behavior, labeled relatively small set of manually labeled data that includes the limbs, and then use that to continue to train the network, and they get pretty good results. I will say what's not shown here is where this is continuing to fail, which is the very, very hard awkward wrestling poses, right? So when it's, I can't even tell where the animals are, and I think it's, if anyone has any ideas for what to do, the animals ride on top of each other in a ball, and you can't actually see any of the limbs, please let me know. Okay. So that's tracking. So what I want to show you now is about the sort of emerging biomedical applications. So once we have these three-dimensional measurements, yes, question.
Yeah, I was just curious. So maybe I missed it, but in your training data set, is it all the same individuals? They the same rats?
For which part? For the one.
The data set you just showed. So when you mark, you annotate.
So the original yellow and red predictions were on individuals not used for training.
Okay.
Yeah. The fully markerless ones, I don't know if those were used for training or not, but certainly it.
Yeah, I was just wondering like how well it generalizes to different rats, basically. That was always an issue.
Yeah, so I think the identification step, like in order for that to be made very robust, what they do is they label those single points within the animals of interest so that you can get the identity track reliably. But after that, it seems to generalize fine to new animals, the 3D part, other than when they're wrestling.
Okay, yeah.
Yes.
Right, for the tracking side, have you tried to use some for the things like keep sort? I mean, is it working? Have you tried it? Yes.
I have not. But for which part?
It's not only based on the bounding blocks, it extracts features per animal, so let's see for algorithms.
But for identifying them. Yeah.
Well, top of feature, one is occluded, they separate from each other, then you're not looking.
So the ID tracking when they're wrestling doesn't seem to be the big issue. It's the 3D pose estimation during the wrestling periods. If that makes sense. I mean, because we painted them, right? So if they leave each other and we miss them, we just find them again. Because it finds the blue one. That makes sense?
I just wanted to know if you use deep sort.
We don't.
Feature extraction, and if it works or not.
We haven't used it. Yeah, we haven't used it. Okay, so that was playing in the background. So, you know, these are the types of waveforms we get for individual animals. You could have seen over here, this is the same animal just from different perspectives. When they're engaged in different behaviors, right, you see different signals within these waveforms, which here are the positions of the individual markers on the body. So, you know, what we set out to do is try to interpret these, try to discover behaviors, try to discover temporal features of behavior that might reveal something interesting and new about, you know, disease states in these animals. And so the first thing that we did, this again was led by Jesse Marshall, who's a postdoc in Solvetsky's lab. He developed that behavioral mapping technique that I just showed briefly beginning in terms of defining the different types of behaviors we included in that RAT7M data set. Here's a little bit more detail on how that's done. So you take in traces of the positions of those markers over time. There's a bunch of different ways to do feature extraction from those traces. Primarily the way we do it, that we get some eigenposture scores over time, which seems to be very effective. We have those little score traces over time, do time frequency transform in order to capture, you know, different frequencies of temporal information over small windows of time that help distinguish individual behaviors that might progress through the same pose in the middle of a particular time window. Okay? Once we have these features extracted, then, oops, you know, we can just TC them or choose your favorite embedding technique. And once you're in that map, then you can cluster that using your favorite clustering algorithm. We did a watershed clustering, the analysis that you've seen here. And then once you've done that, you seem to get this very cool, consistent organization of this map. In this map, put similar behaviors together. That's the whole purpose of this. And of course, those thick black lines are outlining some high-level course behavior, those action categories that we defined in the RAT7M data set. But within each of those course categories, you have these very small, subtle differences between those course behaviors, to this fine level of detail in terms of behavioral identity. And then as I mentioned before, another representation I'd like to use in order to try to interpret the behavior is this ethergram barcode thing. So if you just plot the behavior that the animal is engaged in every frame, plot that over time and color code according to the different type of course behavioral category each of those is in, you get this sort of comprehensive, detailed representation of what the animal is doing over time.
This is the latent string of this thing. What's the behavior of one to one?
Yeah. So any point here means that frame was in that particular cluster. There's 154 clusters for this map. Okay, so here's another view into that. So we have this representation, and what we wanted to do is sort of define new precision units for trying to discover new phenotypes in response to drugs, in response to diseases. And there's the behavioral level, or sort of the body movement level description. Then there's the behavior level description, which is what we've just discussed, so that the animal visits these little discrete behaviors over time. That's what's plotted in ethogram. So each one of those little bars is a different type of behavior over time that the animal is engaged in. What we also notice, though, is that there seem to be these repeated longer timescale patterns of behavioral expression. So it's not just individual behaviors being independent from another behavior over time. These periods of times in which they'll chain together a certain set of behaviors pretty consistently. So you might put a walk, turn, walk set of behaviors together, and that's always there as a little triplet pair. And that might be what we'll call an explore sequence. Or if they do a sniff, walk, active sniff, then maybe that's something they always execute together, and that's an approach sequence. Maybe some sort of eat, delicious ingestion sequence is another type of sequence that might exist in these data, right? So a longer timescale pattern that we can discover in these long longitudinal data sets of 3D pose over time. And then there's potentially another aspect to this hierarchy, which certain types of sequences might be engaged with higher probability when the animal is in a specific type of state, right? Which I think just makes sense from thinking about it. The animal is aroused or hungry, it might execute a bunch of food-seeking behaviors, right, which are embedded within food-seeking sequences. The animal is sleeping, of course, it's going to engage in certain types of behaviors that it wouldn't otherwise engage in the aroused and hungry state. Okay, so we went into the data to see whether or not we could find evidence for these different types of behavioral units, which all might potentially be modulated in response to drugs or in animals in different disease phenotypes, different disease states. So because of time, I'm skipping over a little how we found these things. If we have time at the end, I can come back to it. But let me point out evidence for what we found in terms of sequences and states. So here's another excerpt of an ethogram. And we have this algorithm, this sort of temporal pattern matching algorithm at different timescales. They can automatically find these sequences. So 22 different sequences. So these little short snippets of behaviors over small periods of time. And so that ethogram like this can be converted into this sort of sequence gram, right? As a result of finding these sequences. And a different ethogram that has scale here at 10 minutes or 1 minute. There are these longer time scale periods where the animal seems to be engaged in a particular set of behaviors. So we can also find the states within that period of time. There's usually fewer states and they last longer than these sequences. Here's an example of a sequence versus a state. So sequence 21, which is a left scratching sequence, the animal is always engaged in essentially 5 behaviors all in the same order, right, over time. So it's whatever, scratching itself, moving its leg, sniffing licking its toe, right? And so that's always happening whenever this animal is engaged in this left scratching sequence, which is sequence 21. Whereas a state, right, that we can automatically find has this less defined structure, right? It's just a matter of preferentially executing a certain types of behaviors over a long period of time. Okay. And another way that we can look at these sequences or define these sequences and states are in terms of the percentage of time animals are engaged in one of those course behaviors, right, for a particular sequence. So that left scratching sequence, the course categories that we've defined within that map, animals mostly engaged in scratching type behaviors, and so that's a new representation of what that sequence is, right? So just lots of ledge scratching within that sequence. Kind of hard to see, but you can see there's other bars in the other sequences that define those sequences in terms of the types of behaviors used. The states, on the other hand, look different, have a more homogeneous representation across behaviors, but nevertheless, they can still be defined in terms of the percent behavioral usage across these different behavioral categories. Okay, so given these definitions, can we look at those various levels, see new changes in response to drugs that we couldn't otherwise resolve before? So that was sort of the purpose of the analysis. So this is phenotyping drug effects. What you're seeing here is kind of what we were previously capable of doing in the open field behavior, which is just quantify the amount of time the animal is moving, okay, in response to two different drugs, in this case, caffeine and amphetamine. Just quantify the amount of time the animal is moving, so sort of the coarse automatic metrics that we used to be able to use. You don't see much of a difference. Right? So caffeine and amphetamine seem to increase the activity level of that animal. 2 minutes? Oh gosh, okay. However, if you have access to these more fine-grained behavioral details, you can actually reveal that there's a divergence in terms of the effects of these drugs. The increased levels of activity in response to caffeine and amphetamine, in fact, manifest differently in caffeine versus amphetamine. Caffeine response, the increased activity comes from decreasing the amount of time the animal spent idling, whereas the grooming response seems to increase activity at the expense of grooming for amphetamine. Okay? And then you can also look even finer grained. You can look into these maps and you can see evidence in this little map space of different types of subtypes of behaviors, of these coarse behaviors that change across these different conditions. So amphetamine increases the amount of low rearing behaviors versus high rearing behaviors. And then let me just quickly get through this. Sorry, these plots are annoying to read, but you can also look at the states, right? So I define these states, these longer time scale behavioral patterns. Caffeine and amphetamine have differential effects on these states, so the state level of description. So states that are upregulated in caffeine and not amphetamine. And the states are again defined in terms of the behaviors that are executed in those states. So these states have a lot of rearing behaviors in them over long periods of time. And these states have lots of grooming behaviors in them, and those are things unique now to caffeine in terms of what's brought out. Whereas amphetamine has different sets of states that are upregulated, and those states mostly have to do with walking. So last slide. So based on that analysis, you can find these quantitative differences to visualize whether it's finding anything meaningful in those states and those signals. You could just say, go in and find these states that are very different between those conditions, and then plot the 3D tracking for those particular states. So these are examples of what one might see if you go in and pull out states that are differentially expressed within these different caffeine animal doing lots of rearing behaviors. One's amphetamine animals just turning around in a circle, right? And those are automatically extracted now using the state level analysis of behavior. And finally, we've done something similar for Fragile X. And if anyone went to Josh Wu's poster this morning, I'm sure he'd love to show you some of his videos if you didn't. But he's been doing a bunch of cool work in collaboration with the Tadros lab at Duke to mine a very large data set of Parkinson's mice in response to various treatments. And, still preliminary, but there's some real cool results in terms of behaviors that emerge and which ones are treated by L-DOPA versus other research chemicals lab. Okay, and with that, thanks. Any questions?
Yeah, please come to the podium for a question.
I started late, right?
Yeah, that's fine.
Okay.
Thank you for nice work. I have a quick question. So you use the TC for mapping the behavior states. I was wondering, what if you add new additional mouse data or rat data sets to annotate behavior states? Because TCs are known to be transcriptive method that you need you actually make one model that you need what additional data you actually to run again is needed.
That's
How
one. That's one way to get those points into the t-SNE, but another way is that you can re-embed, right? You can train model to go from points into the map based on the originally mapped data points. And so if you go talk to Josh, he's using some simple k nearest neighbors re-embedding strategy. So he'll fit a map on some conditions, and then he'll take new points and embed them into that map based on the definitions of that space from the original conditions to see how those new points organize within that, the original bounds of that space.
Thank you.
Welcome.
I had a couple questions relating to, well, how do you determine, like, how, first of all, the length of the sequence, and how you can determine whether a sequence is, because sometimes you'll get sequences that you might think be valuable, and then it turns out like they cluster grown, and then you want to determine what's the actually good sequence versus so sequence that you found that may not have any input in terms of drug classification.
So in that particular case, the sequence, well, first of all, there were states. Well, I guess there were sequences in the Fragile X, but I didn't have time to go over them. I mean, you do statistics on the upregulation of the sequences or downregulation, right? So just because you find a sequence doesn't mean it's necessarily different across the experimental conditions, right? So you need to count the number of times that sequence is expressed and then see whether or not there's a statistically significant difference between experimental conditions before you say anything useful about what that sequence is doing.
One quick question. So in the architecture, the input showed you're taking multiple different views and then selecting volume input. I think I saw that you had the 24 cameras or 16 cameras.
Those are motion capture cameras.
So are you using all the views to?
Well, okay. So the 24 was the number of total unique color viewpoints. All the results I've shown you are from 6 views. When we do show markerless data, they come from 6 views. But the more, the better. So it would be nice to use 24. I think like, things get a little cleaner, especially in the hard behaviors.
Go ahead.
Yeah, thanks very much for the talk. I just want to ask you a little bit about whether you're, particularly your sort of latent code used to identify like an anomalous behavior or some event that you can't possibly select for training. So like, I imagine various sort of diagnostically useful signals that maybe for sort of ethical reasons you don't want to collect the training set of them. So yeah, I just wondered if you had anything in that direction that things that don't happen at all.
It's a very important question. I think the jury's still out on how sensitive these analyses are to that type of thing. You should talk to Josh still about it because the Parkinson's data is an example of that. And you know, if you're using a re-embedding technique, you definitely won't discover, you know, those differences, right? But if you embed all of the data together, right, I think in principle, there's nothing stopping the analysis from revealing a new cluster of an anomalous behavior. Well, there's another question about whether or not the tracking is robust to like a drastically different posture that might be induced by a motor disorder. And so we don't have an answer for that either yet. But it's something we're thinking about, and it's a very important point. Thank you. Let's take speaker again. Can you unshare? No, you're gonna share. You have shown, right? So last talk is the oral presentation, and yes, and unless you will present. Are you ready? If you share. Okay. Right. Okay. Hi, everyone. Yeah. Hi, everyone. I'm Anush Kumar. I'm from the University of Minnesota, and I'll also be talking a little bit about mouses. And today I'll be presenting a new data set for mice behaviors called Mouse 3D. So our motivation for presenting this data set was most of the existing data sets are largely constrained to 2D representations, except for a few data sets like PAIR R24M and the RAT7M, like you mentioned. So the constraints with 2D representations are that they are not enough when you're encountering occlusions or when the mouse assumes really complex poses or when their viewpoint changes. For example, in the clip you see here, you can't completely understand the full pose of the mice because of this 2D nature of it. But if you had a 3D representation, it might be more clear what the entire pose the mouse is assuming. So that's our conclusion. And therefore, we decided to build a data set called Mouse 3D, which contains multi-view data, point cloud representations, 3D landmarks, and also a deformable mesh. Yeah. So I'm just going to go over our data collection and data acquisition and data processing so that you guys are more familiar with the contents of our data set. So what we have here is just a small arena with a small tabletop at the center, and it's surrounded by 6 RGBD cameras. The mouse is then placed at the center tabletop and is allowed to roam freely around the environment and explore the environment. And while it does so, we capture the multi-view RGBD data. And using this RGBD data, we then fuse it to form a dense 3D point cloud representation of the mouse, as you can see on the right here, which is a reconstruction of the entire scene. The next task that we do is landmark estimation for the mouse, which is basically estimating 2D key points for the mouse in the RGB images. So we use DeepLab to train a 2D pose estimation model, and then we estimate the 2D key points over all the multi-views. Using this multi-view data, we then triangulate the landmarks and then estimate 3D landmarks for the mouse of interest in this. So previously I showed you the 3D point cloud that we had, but it was a representation of the entire environment. So now that we have the pose estimates of the mouse, we can actually localize the mouse within the point cloud, and we can also extract the mouse from the point cloud. And at this stage, we also assign a coordinate frame to the mouse so that it becomes easier to track the mouse's trajectory later on. So the final thing that we've included in our data set is also a deformable mouse mesh. So what we have is a mesh model which is of 425 vertices, and we actually rig a skeleton to the mouse on Blender, and we extract its rest pose and its skinning weights. So now that we have these two data with us, and we also have the 3D landmarks from the previous steps, we can actually deform the mouse according to the estimated 3D landmarks from our previous steps. So here are a few more examples where the mouse is performing various behaviors, and these are coupled examples of point cloud and meshes. Yeah, so these are some of the numbers of our data set at the moment. Yeah, just to summarize, we are proposing a large-scale multi-view data set which contains RGBD data, 2D key points, 3D landmarks, point cloud data, deform meshes. And over the entire data set, we have about 15 unique mice throughout the data set in the sequences. Yeah, so we think this data set can be super useful when it comes to analyzing mice behaviors, but it can also be used for 3D pose estimation or other tracking analysis as well. Yeah, if you're interested in the data set, please check it out in the website here. And yeah, thank you. Thanks a lot for listening. Okay, so this concludes our CV4Animal workshop in CV4 2022. We hope that we can see you guys next year. And we have 2 hours of a poster session downstairs. please come to them. And I saw a lot of exciting work in the first session, and I'm pretty sure that we'll see another one in the second batch. So I'll see you guys on the first floor. Thank you, everyone.
Thank you so much for awesome organization.
Hey, thank you.
So,